Uh, okay, you can hear me, right? It's a clear. Okay, okay, fine, 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 fine. Is it visible? Okay, 1 minute. And it's moving now. No, it's moving. No, slides are moving. Slides are moving. No, next slide going. No, yeah, fine. Okay then. Yeah, thank you. Yeah.
A pleasant morning to all. Prayer should always be our first response to every situation. I now request Dr. M.G. Daniel Andrew, Assistant Professor, Department of Biotechnology and Bioinformatics, to invoke God's presence.
Shall we close our eyes and bow our heads? Our dear God, our Father, thank you for this second day of the conference, BICPAC. We pray for your grace for the deliberations to go on as planned, for the scheduling, for the participation of people all over India who've registered, for the speakers to be able to convey their thoughts. We pray for your grace on them, and we also pray that our students will gain immense knowledge from this wonderful conference. We pray for all your blessings and mercies today, and we ask you all these things in the matchless name of the Lord and Savior, Jesus Christ. Amen.
Thank you, sir. Now let's begin the plenary lecture of second day session. I invite Dr. R. Sriram, Assistant Professor, Department of Biotechnology and Bioinformatics, to deliver the welcome address and to introduce the respected speaker, Dr. Hitendra Kumar Patel, Principal Scientist, Associate Professor, ACSIR, CSIR, Center for Cell and Molecular Biology, Hyderabad, Telangana.
Yeah, good morning to all the participants. Warm welcome to all for the second day of our international conference. I welcome our honorable speaker of this session, Dr. Hitendra Kumar Patel, Principal Scientist, Associate Professor and CSIR, Center for Cell and Molecular Biology, Hyderabad, Telangana. Welcome you, sir. I welcome convener of this international conference, Dr. V. Violet Dayabaran, ma'am, Head, Department of Biotechnology and Bioinformatics. Welcome you, ma'am. I welcome a co-convener of this international conference, Dr. Jasmine, ma'am, UG Head, Department of Biotechnology and Bioinformatics. Welcome you, ma'am. And I welcome the organizing committee, that is the faculty members of this Department of Biotechnology and Bioinformatics. And I welcome the participant of this international conference. And moreover, and I'm happy to introduce our speaker, Dr. Hitendra Kumar Patel. So in the year of 2002, he did his MSc in Biotechnology. And after MSc in Biotechnology, he has completed his PhD, the International Center for Genetic Engineering and Biotechnology, Italy. And his job title elevated from a junior scientist to the principal scientist between the year 2004 to 2022, which denotes its hard work, dedication, and commitment towards his research. And he has received a CSIR travel grant, and also is a recipient of Arturo Falaschi International Doctoral Fellowship. And he has published more number of research papers
Hello.
So you can begin.
Okay, in between there was some voice break. I'm audible?
Yes, sir.
Okay. So thank you for kind introduction, and thank you for inviting me in this conference. So today I'm going to talk about development of newer rice varieties. And before come to this topic, briefly I'll mention about the place where I'm currently working, and then I'll give some brief background about the work, and then I'll come to this topic. Okay. So this is the main building, CSIR CCMP, where I work. It's in Hyderabad. And we have another campus called Lacoons, or CCMB NX. So this is Laboratory for the Conservation of Endangered Species. And this institute is involved in the conservation of Indian wildlife by using modern biological techniques and assisted reproductive technologies. And then we have another campus called Clinical Research Laboratory. So here we have incubation center and also a lot of work on this COVID. So I'll mention about a few key achievements of CSIR CCMB. Uh, Padma Shri Dr. Lalji Singh, who is the former director of CCMB, he established DNA fingerprinting technology in India. And based on this initiative, later on an institute called Center for DNA Fingerprinting and Diagnostics. Again, this institute is in Hyderabad. So this institute was set up. And so far, more than 20,000 cases related to medical, criminal law, and justice have been dealt by this institute. Also, CCMB developed universal primer technology for species identification. So this is an accurate, and by using this technology, we can quickly identify any particular species. And this technology is patented in 14 countries. So concept here is a small segment of genome can serve as a molecular signature. Okay, and variation in DNA sequence can be used to identify any species, any animal species, bird, fish, reptile, or in mammal species. And use of this technology is in wildlife forensic to stop illegal trade in wildlife and also to detect adulteration of animal meat in food products. And then in Lacoons, we first time in India, the spotted fawn of a spotted deer was developed through artificial insemination. And here we have DNA banking of more than 250 species of mammals, birds, and reptiles of India. And way back in 1990, this company, Shanta Biotech, it was incubated in CCMB, and they developed this hepatitis B vaccine. Also, CCMB is involved in diagnostics of 35 genetic disorders at low price, and also we do genetic counseling. And this is something today I'll speak. CCMB was involved in development of improved Samba Masuri rice. And this rice we developed in collaboration with Indian Institute of Rice Research. So I'll speak about this rice variety. And also since I think graduate and postgraduate students are participating in this conference, so we have skill development program, dissertation program, summer research training program, and also young innovators program. And all these details you can find out at our CCMB website. Okay, so if any information or which kind of research we are doing, all these information are available at our website. So coming to the topic, development of newer rice varieties. Let me give some brief background about the work. So what farmers want? Farmer wants crops with higher yield. They also want crops that are tolerant to biotic stresses so that they spend less money on agrochemicals. They also prefer crops with tolerant to abiotic stresses so that there should not be yield loss because of abiotic stresses, and also crops with better nutrition. Uh, yeah. So ultimately, what we will prefer is a crop which will have all these multiple traits. Okay. So how traits are controlled? Traits are controlled by genes or QTLs, and these trait controlling genes either traits are controlled by monogenic traits, it could be controlled by a single gene, or it could be controlled by multiple genes, which is polygenic traits. And how we can transfer these genes or traits? So there are three ways. One is transgenesis, the second is traditional plant breeding, and third is marker assisted breeding. So what is transgenesis? Transgenesis involves taking an isolated gene of interest, it can be from any source, and modifying it in a tube and reintroducing it into a crop of interest. Okay, provided that tissue culture protocol is established in that crop. So it can be from any gene, from any organism, and we can introduce gene of interest into crop of interest. Okay. So again, these are the steps of transgenesis. We can isolate gene from any source, from bacteria, plant, or any other organism, then clone the gene in a suitable plant expression vector. Then we do transformation, do tissue culture, and finally we select the plant which will have that particular gene of interest. One representative example of transgenesis I have put here, golden rice. You all must have heard about this rice. So this portion, this white rice is a normal rice. Okay. In golden rice 1, what they have overexpressed is carotene desaturase gene from Erwinia iridivora, and also phytoin synthase gene from daffodil. Okay, you can see now rice grains are yellowing, so they now start producing provitamin A, okay, beta carotene. In golden rice too, what they did, that carotene desaturase from same source, but phytoin synthase they have taken from maize, and it makes more amount of provitamin A. So now coming to traditional plant breeding. So how we do traditional plant breeding? We have suppose two plants. One plant is say susceptible for a disease, and other plant is resistant for a disease. So this plant has donor genes or donor QTLs for resistance. We make a cross, we get F1 plant, and then we self to get F2 population. So you consider these are multiple F2 plants, okay, thousands of plants. In traditional plant breeding, we select the plant based on phenotypic selection. Okay, we grow all these 1,000 plants, and then based on the trait of interest, we screen them on the field or greenhouse, and then the plant which have a desirable trait, a phenotype, we select those plants. So what is difference here between transgenesis and traditional plant breeding? In transgenesis, we isolate gene of interest from any source or donor plant, and then we introduce that gene in a plant of interest through tissue culture. So in this case, only a single gene gets transferred. Okay. In traditional plant breeding, what happens? We cross two plants, donor plant and recipient plant. So in this case, not only the gene of interest, but additional regions in the genome also get transferred. So now coming to marker assisted selection or marker assisted breeding. So this is a next step to traditional plant breeding. So consider plant 1 is susceptible for a disease, okay, and here you can see DNA band which has a higher size. Plant 2 is resistant for disease, and here this resistant DNA marker you can see here, and it is lower in size. Okay, in DNA band or gel, you can see it gives lower band. So then when we cross, we get F1, we self, then we get thousands of again F2 population. But in this case, we select the plant based on genotype, not based on phenotype. Okay, we look for this PCR band, and whichever plant will have that resistant PCR band, we only forward those plants. So in this case, we can eliminate lot of work. Okay, we can handle only those plants which have that resistant allele, resistant gene. So with this background, let me come to the topic. So we work on rice, and as you all know, rice is a staple food of India. And in terms of area and production, it is one of the most important crop in India. And here I have put it representative pictures of rice grain. So different part of in different parts, farmers grow different rice varieties. Okay, and one of the popular rice variety is Samba Masuri. It's called BPT 5204. So this is a high yielding rice variety. It is cultivated in about 2 to 4 million hectares, and it has exceptional cooking and eating quality, and also the market price is high for this rice variety, but it is highly susceptible to a disease called bacterial blight disease. Okay, so what is bacterial blight disease? It is a serious disease of rice, and this is the bacteria that cause bacterial leaf blight disease. It is called Xanthomonas oryzae pathwaroryzae. Okay, so this leaf is mock inoculated with water. So what we do, we dip a Caesar in the water or bacterial culture, and then do a clip inoculation. We just cut the leaf. So here this is inoculated with just water, and this leaf is inoculated with the bacterium culture. Okay, and you can see here the disease lesion. It's a yellow in color. In severe infection condition in the field, you can see whole fields looks like a dried up, and there will be serious yield loss. And there are no effective chemicals to control this disease so far. So the best way to control bacterial blight is to develop resistant varieties. Okay, and if we introduce a single resistance gene, pathogen may overcome it, and again it may cause bacterial blight disease. So we should develop a rice variety that has multiple gene of resistance. And in rice, multiple genes for bacterial blight resistance has been already identified. And based on pathotyping study, like which pathotypes are present in India, we selected 3 bacterial blight resistance genes, and that those 3 genes then we introduced into Samba Masuri through marker assisted selection, which I mentioned just now. So this work was done in collaboration with Indian Institute of Rice Research. So what we did, Samba Masuri, which is a fine grain quality, it is high yielding variety, but it is susceptible to bacterial blight disease. Okay. So there was another variety called SS1113. It, it is a poor quality rice. So farmers, they don't prefer to grow this variety, but it has 3 bacterial blight resistance genes, XA21, XA13 and XA5. So we made a cross between these two, and through marker assisted selection, we identified plants that has quality of Samba Masuri, but also that has these 3 bacterial blight resistance genes. And we call that variety as improved Samba Masuri. Okay. So this variety, uh, after development of variety, it was tested through national field trial, and then after successful trial in 2008, the variety got released for and it reached to the farmers. So it is not a transgenic variety. So now you can see the leaf A, it is a Samba Masuri leaf, and you can see disease symptom. This is a typical bacterial blight disease symptom. And leaf B is improved Samba Masuri, and there is no disease symptom. Okay, it's not moving here from say zero point 0.5 centimeter, and this is in one of the field station. So this crop is improved Samba Masuri, it is healthy, and this is Samba Masuri. You can see it is affected by bacterial blight disease. And this is a picture in the farmer's field. Again, Samba Masuri is affected by bacterial blight disease, and improved Samba Masuri is free from the disease. Here you can clearly see Samba Masuri field, again severely affected by bacterial blight disease, and improved Samba Masuri is free from the disease. Also, we check the grain quality and cooking quality, and improved Samba Masuri has similar grain and cooking quality, and this is important. Otherwise, if there is any alteration, farmers and consumers, they will not accept the variety. So then we have this program, it's called Blight Out program of CSIR. And in this project, we popularized this improved Samba Masuri rice, and we distributed 10 kg seeds per farmer, which is sufficient for 1 acre area. And so far, we have distributed to about 10,000 farmers in the states of Telangana, Andhra Pradesh, Tamil Nadu, Karnataka, Chhattisgarh, Jharkhand, Uttar Pradesh, and a few other states. And these are, I'll show few field farmers day pictures. So this is from Nadupatti village near Pulithalai in Karur district. So we visited this place in 2017. We interacted with the local farmers and agriculture officials, and this crop is improved Samba Masuri. Also in Uttar Pradesh, we distributed seed. He is Agriculture Minister of Uttar Pradesh. So like that, we distributed improved Samba Masuri seeds to different states, and we popularized the variety. So, so far this variety has been grown in about 7 lakh hectares in the states of Telangana, Andhra Pradesh, Tamil Nadu, Karnataka, Chhattisgarh, Uttar Pradesh, and other states. And currently, the annual cultivation of this variety is about 2 to 3 lakh hectares. And what impact the variety has made? So total value of ISM produce is estimated to be about 6,700 crores, and out of which the benefit to farmers because of bacterial blight resistance is close to 1,200 crores. Okay, and this projection is based on impact analysis done by Manage Hyderabad. So this variety is becoming popular among farmers and spreading from farmers to farmers. And so far we have licensed this variety to 3 companies. In 2011 to Sri Biotech, in 2015 to Metahelix Life Sciences, which is a Tata Group company. And these 2 licenses were for seed, like to sell improved Samba Masuri seed uh, as a bacterial blight resistant variety. And recently we licensed to Sri Krishna Rice Mill for improved Samba Masuri grain as a diabetic friendly rice, which I'll come to the how it is diabetic friendly. Okay, and for this development of variety, so far we have received 2 awards, one from CSIR and one from DBT. And so recently we tested this rice for its glycemic index value. Okay, and this test was done by ICMR, National Institute of Nutrition, and based on human feeding trials, they reported that improved Samba Masuri rice has glycemic index value of 50.99, and foods with glycemic index value that has below 55, those foods are considered good for diabetics. Okay, because in these foods the glucose is released relatively slowly in their bloodstream after consumption of food. So what is glycemic index? Glycemic index is ranking of carbohydrates on a scale from 0 to 100 according to the extent of which they raise blood sugar or glucose levels after eating that particular food. Okay, so when we eat foods with a high GI, they are rapidly digested, absorbed, and metabolized, and because of that, there will be sudden fluctuation in the blood sugar or glucose level. And if we eat low GI foods, then there will be only smaller fluctuation in the blood glucose levels. So low GI foods are good for long-term health, reducing risk of type 2 diabetes and heart disease. and also it is also one of the keys to maintaining weight loss. So as we can see here, if we monitor the blood glucose level for 2 hours, if high GI food uh, profile we see, there will be sudden increase in the blood glucose level, and if we eat low GI food, it will not be sudden increase. So to summarize the improved Samba Masuri story, this variety was developed around 2 decades ago, and finally it was released in 2008. And so far we have licensed the variety to 3 companies, and we have received 2 awards. And this variety is a low glycemic index variety, a diabetic friendly rice. And this was clinically tested by NIN in 2017. And through CSIR 800 Blight Out program, so far we have distributed seeds of this rice variety to about 10,000 farmers in different states. So what next after improved Samba Masuri? Okay, improved Samba Masuri is resistant for bacterial blight disease, but it is susceptible to many other, still susceptible to many other diseases based on abiotic stresses. And again, based on farmers' need, we need to fortify these additional traits. Okay. Okay. So now what we are doing, because improved Samba Masuri, uh, we need to improve for other traits. Okay, other, so like blast, it is another fungal disease, and brown plant hopper. This is an insect pest that cause damage in the rice. So these are two biotic stresses. And again, we are adding genes related to tolerance to these biotic stresses into improved Samba Masuri through marker-assisted selection. There are many abiotic stresses like drought, lodging, submergence, salinity. And again, genes and QTLs for these traits are well known, and we are now adding into improved Samba Masuri. And also these two genes we are introducing to increase its yield. Okay, so even it is high yielding variety, we would like to further increase its yield so that farmers can get benefit. So one by one we are doing this, and ultimately we'll have kind of super improved uh, Samba Masuri rice, which will have these multiple traits. And first success in this effort is we have developed a variety in improved Samba Masuri background that has salinity tolerance. Okay, so saltol QTL was introduced into improved Samba Masuri. So this is improved Samba Masuri plant, and this is the donor line from where we have introduced that saltol QTL. And these four are improved Samba Masuri introgressed lines. Okay, as you can see that they have better survival under salt stress, and also they have better root system compared to Samba, improved Samba Masuri. So it has already passed the national field trials. For any variety to reach to farmers, that national field trial is must. Okay, so it has already passed national field trials, and recently it was released, approved for release as a variety in coastal salinity zones. Here I have put some representative pictures of panicle, and in this case we have introduced high yielding related gene, and you can see the number of grains are more compared to input Samba Masuri. And here we have introduced two genes, one for high yielding and one for stronger culp. So here the thickness of stem will be more, and it will not lodge. Okay, it will give high yield. And this variety, based on yield improvement, already promoted to advanced varietal trial too. Okay, and we are hopeful that it will pass the national field trial and again reach to the farmers. So what we have seen that marker assisted selection works. Okay, we are introducing multiple genes through marker assisted selection, but marker assisted selection can only be applied to those traits for which the variation is already available in the gene pool of price. Okay, and in this case, then can we extend the scope of marker assisted selection to provide solution for traits that are not available in the gene pool of rice? So which are not available in wild rice varieties or any rice varieties. So considering that, what we are doing is we are trying to now identify novel genes or QTLs that are associated with important agronomic traits. And in this work, what we have done, we have developed 10,000 mutant population. So this is mutagenesis through EMS, which is a chemical mutagen. So we developed 10,000 lines, and then we have screened these mutant population for agronomically important traits. Okay. Uh, so here you can consider seeds are treated with EMS. We get M1 plants, and then at each stage M through M2, M3, M4, like that we screen for the traits, and currently we are at M7 stage. So what traits we have screened? We screen for bacterial blight resistance. Okay, why we did this? Because suppose down the line, improved Samba Masuri resistance in improved Samba Masuri is break by evolution in the pathogen. Then we may need newer source of resistance. So then in that case, the mutant line that is resistant for bacterial blight disease, we can use as a donor. So with that objective, we screen the mutant population for bacterial blight resistance. And here you can see this is a Samba Masuri leaf, and it is, you can see clearly, you can see bacterial blight disease symptom. And these are two mutant lines where we can see they are resistant to bacterial blight disease. Yellow stem borer is a serious insect pest of rice. This is how it looks like, and it causes dead heart symptom in young vegetative stage, and also it causes white ear symptom in the reproductive stage. So here in this case you can see white panicles, so they are empty seeds, and because of this farmers will have yield loss. And so far there are no known source of tolerance available in the rice germplasm for yellow stem borer. So whatever will tolerant mutant line we get will be novel. Okay. So with that idea, we screen the population, and again you can see this is Samba Masuri line, and you can see damage, dead heart damage, and this is one of the tolerant mutant line, and they show no symptom of YSB. Also at the reproductive stage, this is Samba Masuri line. This is another susceptible line. You can see white ear symptom, and this is one of the tolerant line, and there is no white ear symptom in this tolerant line. Sheet blight is a serious fungal disease of rice. You can see this gray spot, and it is caused by a fungal pathogen Rhizoctonia solani. And again, there are only a very few reports of tolerance of sheet blight. There is a variety called Tetup, which show moderate level of tolerance. And again, we screen the mutant population and identified mutant lines that show good level of tolerance. So this is a susceptible Samba Masuri rice, and this is a mutant plant. You can see looks healthy here. And again, lodging is a big problem, particularly in coastal areas, because of cyclonic condition. And you can see the thickness of stem of Samba Masuri, and this is one of the mutant line. Thickness is almost double. So in this case, plant will be more sturdy, and it will not fall to the ground. Also, we have identified mutant lines that show early maturation. Okay, you can see this group of plants. So they have already flowered, matured, and this is Samba Masuri. There is no panicle visible here. So this will be helpful for farmers like they need less water or less fertilizer. If duration is less, farmers can take multiple crop in a year. Also, Samba Masuri has issue with the emergence of panicle. There is panicle choking, and because of this, there is yield loss 5 to 10%. And we have identified mutant lines where panicle emerged completely, so then we can minimize this yield loss. And also, we have identified several mutant lines that show high yielding phenotype, like panicle length is more, number of grains are more, so like that. And some of these lines are currently in national field trials. Also, we are working towards nutritional related traits, and we are trying to identify mutant lines that show enhanced level of iron, zinc, and protein. So in our ongoing work, we are trying to develop markers for these important agronomic traits. Like we are trying to identify which region is responsible for those phenotype in the mutants, and this we are doing through genetic methodologies, next generation sequencing, and bioinformatic analysis. And once we identify the marker, we develop the marker. Through marker assisted selection, then we can use uh, uh, these mutants and markers together as a technology package, and we can introduce these important agronomic traits into improved Samba Masuri rice or any other commercially important rice varieties. Yeah, thank you. And I would like to acknowledge Dr. Ramesh Sonti. So he initiated all these rice work. He and Dr. Sundaram from Indian Institute of Rice Research, their team jointly developed improved Samba Masuri rice. And I would like to acknowledge all other lab members. Then IIR team, particularly Dr. Sundaram, who is Director of Indian Institute of Rice Research, and Dr. Seshu Madhav, who is helping in characterization of mutant lines, and all other team members. And we have funding from CSIR and ICAR, and I would like to thank directors of CCMB and IIR. Yeah, so if anybody is interested in licensing this variety or selling this improved Samba Masuri as a diabetic friendly rice, they can reach to this email BDG. It's a business development group @ ccmb.press.in. Okay, thank you. I'm open for any question.
Thank you very much for your thoughtful lecture, sir. Now the session is open for discussion. Dear students and scholars, you can post your questions. Kindly unmute your audio and you can post your questions.
Yes, anybody has any question? Yes, Akshita.
Good morning, sir. I just wanted to know whether there has been any clinical trials that has been conducted on diabetes subjects and their glucose levels have been checked post giving this type of cereal.
Yeah, so particularly for improved Samba Masuri, this clinical test we have done at NIN. Okay, it's a National Institute of Nutrition.
Yes, sir. I am aware of it.
Yeah, so what they did, they feed to 10 human volunteers.
Okay.
And then they monitored their glucose level after 2 hours of feeding this rice.
Okay.
And based on average of those 10 human volunteers, then they reported that ISM is a low GI rice and GI value is 50.99.
Okay, sir. Is there any paper published on?
On low GI, we haven't published yet.
Okay.
I think soon it will be there. But
Okay.
development of variety, yes, improve how this variety was developed. It was published in 2017 in New Phytica.
Okay. All right, sir. Yes. Thank you so much, sir.
Sir, I have a question. Your presentation was very wonderful.
Okay.
Sir, related to this rice mutant variety, Masuri, how long it takes the period to obtain this mutant variety?
Actually, it all depends. Like development, maybe you will develop in 1 year. Okay.
Okay, sir.
Then screening, you have to do 3-4 generations so that that trait is stabilized. Okay, it should not segregate. And
Okay.
once you are confident of a line, so say it will take 2 years to stabilize the trait, then again it should go to national field trial, which is another 3 years. So
So, a
altogether,
long time research work is necessary
yeah,
to.
so altogether a variety to reach to farmer, minimum it takes 5 to 7 years.
Okay, sir. Thank you for a very wonderful, extensive work. And hope so we can also do practice in other varieties of rice also.
Right, right.
Okay, sir. Thank you, sir. Thank you.
Okay, thank you. Is there any other question? So can I stop sharing now?
Is there any more queries? Dear participants? Thank you very much, sir, for your wonderful presentation and talk. Thank you, sir.
Thank you. Thank you. Thanks again. Okay then.
Thank you, sir.
Yeah. Okay. Bye-bye.
Now let's move on to the next plenary lecture. I invite Dr. M. Vinit, Assistant Professor, Department of Biotechnology and Bioinformatics, to introduce the respected speaker, Dr. Gopalu Karnagaran, Research Professor, Department of Fine Chemistry, Seoul National University of Science and Technology, Seoul, Korea.
Good morning to all. Now I am introduced a special talk, Dr. Kobala Krishnan, Research Professor, the Department of Fine Chemistry, Seoul National University of Science and Technology, Korea. Dr. Gopalakrishnan acquired his Master of Philosophy in Biotechnology from Bardiyar University, Coimbatore, 2010, and joined as Junior Research Fellow in the Center of Nanoscience and Technology, KS Rangasamy College of Technology, Tiruchenkodu, Tamil Nadu. He received his PhD degree in 2014 from Anna University and joined as assistant professor in the Department of Biotechnology at KSR College of Arts and Science, Tiruchenkodu, Tamil Nadu. In 2015, he has selected as Young Scientist Fellow in the Department of Functional Nano Systems and High Temperatures Materials, National University of Science and Technology, Mexico, Russian Federation. In 2017, he served as visiting professor in the Department of Functional Nanosystem and High Temperature Materials, National University of Science and Technology, Mexico, Russian Federations. In 2018, he received the Korean Research Fellow in the Department of Fine Chemistry, Seoul National University of Science and Technology, Seoul Republic of Korea. In 2021, he has been promoted as Brain Pool Fellow in the Department of Fine Chemistry, Seoul National University of Science and Technology, Seoul Republic of Korea. His research interest focus on synthesis of nanoparticles using plants and microbes, characterization and applications of nanoparticles in biomedicine, studying magnetic property of nanoparticles and nanocomposites. Presently, he is working on nano hydroxypeptide from natural research of bone implement and wood healing applications. He has received many awards such as Young Scientist Award in 2015, Korea Research Fellow Award in 2018, and Brainpool Fellow Award 2021. He served as principal investigator of one completed sponsor research project worth 17 lakhs from Ministry of Education of Science of the Russian Federation. At present, one sponsor research project in the turn in 93 lakhs in under the progress in the sponsorship from Basic Science Research Program through the National Research Foundation of Korea, funded by the Ministry of Education, Republic of Korea. His passion towards research lead him the public from the 94 research paper in reputed international journals with a total citation of 1985. As per Google Scholar, one citation report and 14 paper in conference proceeding. He acted guest editor of 3 international journals and presently reviewer in more than 30 international peer-reviewed journals. He is a fellow and life members in different scientific society, both in India and abroad. So thank you, sir. Welcome.
Hello. Hello.
Hello, yes, sir.
Hello. Hello. Hello, ma'am. Can you hear me?
Yes, sir. You're audible.
Yeah.
Sir.
Yeah. Okay. Thank you. Thank you for a nice introduction by Dr. Vineet. So, am I audible to everyone? Can everyone listen me? Please respond so that I can understand that everyone is listening me properly.
Yes, sir.
Yeah. Okay.
Messages from the chat box.
Okay, ma'am. Thank you, ma'am. Thank you for inviting me. So today I'm here to present one of my research topic, eco-friendly metal oxide nanoparticle synthesis towards effective cancer treatment. So basically, this work is based on nanotechnology. So I know maybe you are not aware of this nanotechnology, but I will try to make you some familiarity about this research work. So in this presentation, I will give you some detail like introduction, objective, and review of literature and methodologies, what we have performed, and the results and discussion about the works we have performed. And then it will be based on the summary and conclusion, and then acknowledgement. So basically, what is cancer? So cancer is like a most deadly disease which occurs due to the continuous cell growth. So for example, every cell has a lifespan, like it will initially it will multiply and then it will grow and then it has a lifestyle of death, cell death. But generally in cancer, so there will be no cell death kind of thing. So it will just move on, multiply, and then there will not be any like cell death. So WHO estimated like 1.7 million of new cases of lung cancers every year, and estimate death is more than 60 lakhs in 2021, and still the number of cancer cell is increasing day by day. So for cancer treatment, generally there are different methods like radiation, chemotherapy, and surgery as a major treatment methods nowadays is available. But for to overcome such a treatment method, nanotechnology has introduced some techniques and some materials which can be used for the treatment of cancer. So nanoparticles are actively developing targeted based tumor development and treatment for that. So in that particularly zinc oxide is one of the forecasting inorganic compound in the area of science and technology, which has been proven for the treatment of cancer. So in order to modify such zinc oxide, we have developed few techniques. So and then this work was published in Applied Organometallic Chemistry with impact factor of 4.1. So this is the paper which I am going to share with all students and members to know and get familiarity with zinc oxide. So generally, for every research, you know that there are some kind of objectives to be performed. So the first objective was to synthesize zinc oxide nanorods by using Cycas plant leaf extract, and then to characterize it and then evaluate the safety of zinc oxide nanoparticles, and then to determine the total antioxidant activity and to investigate the anti-cancer effect of zinc oxide nanorods. So what is like lung cancer? So lung cancer is generally being introduced because of the regular intake of tobacco and consumptions and all in man and women. Then cancer targeted therapy using nano-based medicine have been attracted many researchers in the present day. Different metal oxide nanoparticles are studied and reported, but zinc oxide is having a unique property. And for metal oxide nanoparticle synthesis, there are different methods like sol gel method, thermal evaporation method, mechanical milling, hydrothermal synthesis are available to synthesize nanorods. But these methods are very expensive and require a lot of toxic materials, and also it requires a lot of time consuming. To overcome such difficulties, plant mediated synthesis method is considerably as an interesting approach in recent times. Until today, various biological materials such as fungus, bacteria, plants have been used for the synthesis of nanoparticles. However, plant is the cheapest and abundant in nature and can be utilized for large scale production. So that is why we have chosen the plant extract for the synthesis. So generally now I will go to some basics like what is called synthesis. Like it is like a production. So for example, you have hello.
Sir,
hello?
excuse me, sir.
Yes, hello.
Sir, I'm Vinit, sir.
Yeah, sir. Tell me, sir.
Sir, please kindly share for your PPT, sir.
Okay, okay.
Sir, PPT is visible, sir.
Okay, sir. Sorry.
Sir, thank you. Sorry for the disturbance, sir. Please, sir.
Okay, sir.
Thank you very much, sir.
So sorry, you could have informed me earlier. So
Sir, PPT is visible, sir.
okay.
Thank you very much, sir. Please continue for your talk, sir.
Okay, sir. Wait, sir.
Sorry for the disturbance, sir.
It's okay, sir. But you would have told me. So
Okay,
earlier,
it's okay, it's okay, sir.
it's okay. Is it viewable, the video? My presentation, is it? Can you see my video? Hello? 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 Hello, ma'am. Hello. Anybody can see? Can you see my presentation? Hello? 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 Can you hear me now? Hello, please respond somebody so that I can know that you are viewing my presentation. Hello? 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 So now I will continue. Sorry for the disturbance about the issues of sharing the screen. So now in this work, I will just share you some information like how we synthesized and how we produce our nanoparticles and how we used for checking of anti-cancer activity. So generally we took a cycas plant from the Andaman Nicobar Island, and then we collected it, and then we have prepared the plant extract, and then we used zinc acetate and we mixed with the plant extract, and we synthesized the zinc oxide nanorods. And these nanorods were characterized by using different techniques like XRD, FTIR, SEM-IDAX, TEM, BET for the confirmation of produced zinc oxide. So then we have analyzed the antioxidant activity by DPPH method. Anti-cancer activity. We have used the in vivo protocol for for anti-cancer activity. We used in vitro method by using MTT assay and as XZT assay, and then NRU assay, and then LDH assay to understand the anti-cancer activity. And for in vivo analysis, we used a zebrafish embryo, and then for xenotoxicity assay, we used the COMET assay. And for oxidative stress parameter, we measured the ROS and then lipid peroxidation assay. So generally, in nanoparticle synthesis, there are two approaches. Like one is bottom-up approach and one is top-to-bottom approach. So in this method, we use like bottom-up approach. So what is bottom-up approach is like the metals ions will be in the ionic form, and this ionic will be combined in by small combination of these ions into atoms, and then these atoms will be converted into the nanoparticles. So we use cycas plant extract. So you can see that this plant extract been mixed with zinc acetate to form the zinc oxide nanoparticles. So in this, what will happen? The biomolecule will be used to act as a reducer. So the zinc acetate will be converted into zinc oxide nanoparticles. So this is the method. This is a very simple, easy method for the synthesis. So in this characterization, the nanoparticle which we have obtained from the plant extract is been characterized by different techniques like XRD, FTIR, SEM, then TEM and BT analysis. So in XRD, the XRD is a technique in which we will get the spectrum of the nanoparticle and which is matched with the standard material. So we have obtained that the standard material zinc oxide is exactly matching with the material which we synthesize, and the functional group was identified by using FTIR spectrum, and then the sizes were identified by SEM and TEM analysis, and the surface area of the nanoparticle is being identified by BET surface area analysis, and the antioxidant is been identified by DPPH method. So we confirm that the zinc oxide nanorods has been successfully synthesized by the using of plant extract. So in first chapter conclusion, so the plant extract were synthesized, and then it was confirmed to be crystalline, hexagonal crystalline with JCPDS9 number is 0760704, and the functional groups were identified to have the functional groups of aromatic, ether, dimer groups were present in the zinc oxide nanoparticles, and the EDX spectrum has confirmed that the presence of zinc and oxygen was present, and SEM and TEM analysis confirmed that the size of the nanoparticle is between 20 to 200 nanometer, and it is of rod shaped with an average particle size of 150 nanometer. And then zinc oxide nanoparticle surface area was found to be 25 meters square. gram, and with the help of BT plot. And the zinc oxide nanoparticles have an excellent scavenging activity. So this was the first chapter. Then in vivo analysis, we used a zebrafish to confirm that the zinc oxide which we produced is not toxic. So we used like embryo, zebrafish embryo, and we treated with different concentrations of our synthesized nanoparticles. And we confirmed that this synthesized nanoparticle is non-toxic to the zebrafish. So then we have, as I told you earlier, like we have evaluated the anti-cancer activity by using different standard methods like anti-T, XTT, and NRU and LDH method. These are the basic methods which we use for the confirmation, like to know whether these synthesized nanoparticles have some anti-cancer activity. And we observed that it has an excellent anti-cancer activity by using these techniques. And in conclusion, in vivo toxicity evaluation confirms that the synthesized zinc oxide nanorods are safe to use. MTT, XTT, NRU, and LDH also confirm that these have an anti-cancer properties. So generally, the anti-cancer activity have different mechanisms. Like you can see here. So there are certain mechanisms being found by different various researchers, and so how it works, like you can see that these are zinc oxide nanoparticles. So when it comes in contact with the cells of the cancer cells, so it will produce the ROS, the reactive oxygen species being generated because of the activity of this nanoparticle. It interact with the surface of the cells, and sometimes it directly damages the cell membrane, and in some other cases it also damages the DNA and it arrests the cell cycle. In another cases, these nanoparticles arrest the protein inhibition and leads to the cancer cell death. So after this cancer death, there will be a release of zinc ions. So these are the possible induced mechanism by which these zinc oxide nanoparticles are making anti-cancer effect. So to confirm the xenotoxicity effect of our synthesized nanoparticle, we used two different techniques, like a percentage DNA method and Oliver tail method. So in these two methods, we confirm that our synthesized zinc oxide nanoparticle have an excellent xenotoxicity effect also towards the cancer cell lines. And so in conclusion, zinc oxide nanorods showed 35% of oil tail migration, and zinc oxide nanoparticle exposed to say A549 cells shows 45% of intracellular ROS development. And these cells have a higher lipid peroxidation level. So because of that, it has an anti-cancer effect. So in the next slide, I will show you that there are certain molecular mechanisms, which is involved during the zinc oxide. So you can see here, it occurs due to the apoptosis mechanism. So apoptosis occur inside the living cell with a series of molecular mechanisms which are based on protein signaling, MAPK pathways. So these MAPK pathways have three main pathways. Like one is stress activated protein kinase, then second one is extracellular signaling kinase, third one is Jun and terminal kinase, and P38. So these are the three major pathways by which the zinc oxide nanoparticle introduced an apoptosis mechanism inside the cancer cells, leading to the cell death. So you can see here, when zinc oxide is introduced in the living cells, so there are three pathways, 1, 2, and 3. These three pathways are the major pathways of MAPK pathway, which leads to the apoptosis of the cells. So in summary and conclusion, so what we have done is, in the basic, what I'm telling you, like the zinc acetate precursor was transformed into zinc oxide nanorods with the use of plant extract. the plant extract converted the zinc acetate into the zinc oxide nanorods. And then we confirm by XRD that it is highly crystalline material. And by FTR functional group analysis, we confirm that biomolecules were introduced in this synthesis mechanism, and it converted the zinc acetate into a nanoparticles. And then the surface area was confirmed by BET analysis. Then SEM and TEM are the two techniques which confirms that these are nanorods-like structure with less aggregation. And EDX is a technique which confirms the elemental presence. So the elemental presence was confirmed by the EDX analysis, and it shows that the presence of zinc and oxygen was present. And then the antioxidant confirms that our synthesized zinc oxide nanorods have an excellent antioxidant activity, and it was based on time and dose response. And zinc oxide nanorods doesn't have any mortality change in the zebrafish, which confirms that this is safe to the human. Then the anti-cancer activity also confirms that this has a very good anti-cancer activity against lung cancer cell lines, and which is confirmed by COMET assay and oxidative stress assay also. And the mechanism of zinc oxide nanoparticles activity was confirmed by oxidative stress-induced DNA damage followed by apoptosis. These studies will be definitely very useful for the as a nanomedicine for the human lung cancer treatment in a eco-friendly method. So this is just was the studies what we performed, and I would like to thank my PhD supervisor, who like Dr. Venkachalam Rajendran, who has guided me for my PhD degree, and I would like to thank postdoctoral supervisor in Boom Cho for supporting my research work. I would like to thank the funding agency like National Research Foundation of Korea and Ministry of ICT for successful completion of this research work. I would like to thank Seoul Tech head, professor, postdoc, students, and all technical staffs who are behind my research work. I would like to thank the organizing committee, Department of Biotechnology and Bioinformatics, Bishop Heber College, Trichy, for inviting me and giving this opportunity to talk in front of all students and faculty members. And at last, I would like to thank all my family members and friends for their continuous support. And thank you. If you have any doubts, you can ask me regarding my presentation. I know it was little tough for the students because of some technical issues, but I hope you have understand few things from this presentation. So now the session is open for the discussion and queries. If you have, please ask me.
Dear participants, your times, please ask any questions. Dr. Kanan, sir.
Yes.
Sir, thank you very much for the wonderful topic, the nanoparticle synthesis.
Yeah, thank you.
It's very informative. Thank you, sir. Thank you for your valuable timing to accepting our international conference. Thank you very much, sir.
Thank you, sir. Thank you. Sorry for the technical issue during my presentation. Sorry.
So it's okay, sir. This is no problem, sir. Thank you for your valuable time, sir. Thank you, sir.
Thank you.
Let's all have a break for tea and refreshments.
TV.
All kindly join by 10:15 p.m. for the next lecture.
A very good morning to all the participants. We are about to start the next session. So this will be a poster and oral presentation. So in the general link, we have the participants of group 1 poster presentation. The other participants will join in their respective link already posted in the WhatsApp groups. So I request the participants to join in the respective Google link, which has been posted in the WhatsApp group. Let me read out the group 1 poster presentation members who will be joining in the general Zoom link to present their posters. Participant number 1, Shweta Kashyap. Number 2, Jabina. Number 3, Sumati and Vimal Priya. Number 4, Man Deepika. Number 5, Jayalakshmi. Number 6, Seena. Number 7, Anju Kumari. Number 8, Pooja Tak. Number 9, Ramya. Number 10, Tara Paul. Number 11, Uma Maheshwari. Number 12, Murugash. Number 13, Kumar Amit. Number 14, Shruti Yalani. Hope these members are ready with your presentation. Please be ready with your slides. Once we call out your names, please raise your hand so that we will unmute you and you can at once start your presentation. So thank you for your cooperation. We shall now proceed with group 1 poster presentation. Dear participants, could you hear me? You can type in the chat box. Dear participants, are you able to hear me? The first participant, Shweta Kashyap. If you're ready, serial number 1, Shweta Kashyap. Kindly respond. Kindly raise your hand so that we can unmute you. அவங்கள டெக் பண்ணவா? பூஜா தக்கு வந்திருக்காங்க. ஒரு நிமிஷம் இருங்க, நான் டெக் பண்ணிக்கிறேன். we move on to the poster presentation. May I request Vimal Priya to present her poster? Vimal Priya, are you able to hear me?
Yes, ma'am.
Yes, please proceed with your poster presentation.
My slide is visible, ma'am.
Yes, it is visible. Please proceed.
Okay. Good morning, one and all present here. I am Vimal Priya. I am studying as a PhD research scholar at Kongunadu Arts and Science College. This is my previous college postgraduate project work, that is development of starch-based biodegradable bioplastic from seeds of Amaranthus cruentus under the guidance of Dr. R. Sumati in PSGR Krishnamal College. Okay. Introduction. Biodegradable plastic. We all are known what is biodegradable plastic. These are biologically degradable. Most of the scientists are focused to find out which one source is the best for production of the biodegradable plastic in nowadays. Most of the several sources are there. That is a microbial source, plant source, agricultural waste source. Here we use a plant source for the production of biodegradable plastic. It is eco-friendly and easily decomposed by the microorganism. These are carry bags, garbage bags. These are some products synthesized by biodegradable plastic. Okay, this is the reason for why we choose the Amaranthus cruentus for the synthesis of biodegradable plastic. The Amaranthaceae family plant seeds have a unique starch itself. It have high nutritional value. Amaranthus cruentus is one of the species of the family. Comparing to other seeds like wheat, rice, corn, its size is very minor, but it have 90% of the starch from its total weight. Okay. Another advantages of the starch is that have a high amylopectin content, that is 88 to 99%, and thus makes it a waxy type of the starch with some unique characteristic such as high viscosity, gelatinization at the higher temperature. The main objective of the study is first is extraction of the starch and then production of the plastic material from the starch. Then after acid test, alkali test, solubility test, flame test was conducted for to check the biodegradability of the synthesized plastic. Next one is the detection of the different compound using the FTIR method. These are materials used for our study. Amaranthus cruentus seeds, distilled water, sodium hydroxide solution for alkali strip method, and hydrochloric acid for pH adjustment, and glycerol gelatinization, gelatin for use for the plasticizer, and acetic acid, sulfuric acid. Equipment used were weighing balance, blenders, centrifuge tube, oven, hot plate, pH meter, FTIR analysis. This is the overall procedure. First, we collect the plant material and remove the seeds from the plant. After the seeds was used for the extraction of the starch, extraction of starch, and the starch was extracted by centrifuge method, and the extracted starch was dried in a hot air oven. After the starch was used for the synthesis of biodegradable plastic. First, we take a extracted starch in a beaker and add a sufficient amount of water in that, and add a regular amount of glycerol and gelatin, and check the pH and adjust the pH neutral, because that only ideal for the synthesis of biodegradable plastic. After the mixture was heat to make a slurry, the slurry was poured on the aluminum foil. Then the aluminum foil is kept under the incubator at 40 degrees Celsius. Allow the material for 2 days. Sorry, ma'am. 2 days. After 2 days, we scrapped out the bioplastic material from the aluminum foil. The production biofilm was characterization by acid test, alkali test, and solubility test, flame test. This was conducted for the biodegradability capacity of the synthesis material. After the biofilm is detect the different compound using the FTIR. These are results. The alkaline test was done with the sodium hydroxide. The time taken for the bioplastic to dissolve the sodium hydroxide is 30 minutes. Here I didn't mention hydrochloric acid test and time taken for dissolving the bioplastic material in the hydrochloric acid is 220 minutes. The solubility test was done in the distilled water. The time taken for the bioplastic of the dissolved in the distilled water is 125 minutes. So the bioplastic is sensitive to alkali solution. Okay. The flame test was conducted and the time taken for the bioplastic from ash is 2 minutes. So these are concluded that our synthesized plastic material is biologically broken. This is a very easy and take a small time only. Okay. The next FTR analysis also performed for characterization of some peaks showing the presence of the functional group like aromatic hydrocarbon, alpha beta unsaturated stretching, then C double bond C, C double bond N. These are some active compounds present in our bioplastic material. The present study was concluded that the synthesis of starch-based bioplastic from the Amaranthus cruentus seed starch was a feasible solution it is a best substitute for the petroleum-based plastic, as they were prepared from merely natural source, making them being decomposed more easily by microbes and therefore environment friendly. Conclusion of my study: Amaranthus cruentus seed starch would be an effective substrate for the production of starch-based bioplastic. The properties of the film are influenced by the composition of the flour mixture and glycerol concentration. The Amaranthus cruentus seed starch contribute to the strength and elongation of the film as a result of its higher content in the amylose. It is also found that the higher concentration of the glycerol will result the lower value in the tensile strength and higher value of the elongation and break. The synthesized starch-based bioplastic film exhibit the good mechanical properties and are excellent oxygen barriers. Thank you. Ma'am, I'm complete, ma'am.
Can you share your slide once again, your poster? Yeah, it takes 100 and it takes 125 minutes to degrade. So what will be the strength or the durability of the bioplastic that you're creating? If it can it be used? So for what purpose you have created this bioplastic? Because you have mentioned it can be used for carry bags, garbage bags, water bottles, and spoons. So that is what plastics have been commonly used nowadays. But your material, what is the scope of your work? For which purpose you are developing this bioplastic?
Ma'am, nowadays most of the food materials are wrapped by the plastic only, ma'am. So this kinds of bioplastic is helpful to maintain the food quality, ma'am. So
okay, but you say that it gets dissolved in the distilled water within 125 minutes.
Yes, ma'am.
So if you wrap it in a food thing, how long it will withstand? That's what I'm asking.
Ma'am, can we modify the this character by
so it's in your primitive work you are doing. So later I think more work is needed. Okay, thank you, Vimal Priya, for your presentation. Can we have Deepika as the next presenter? Deepika, can you please raise your hand? Deepika. Deepika Mann, are you ready with your poster? Can you hear me?
Yes, ma'am.
Yes, please present your poster.
Okay, ma'am. Ma'am, is it visible?
Yes, it is visible. Please go ahead.
Okay. A very good morning to all the faculty members, Bishop Harbour College, organizing team, and co-participants. Today, I, Deepika Mann, Research Scholar, Department of Geology, Kurukshetra University, Kurukshetra, Haryana, feeling privileged to present my poster in this international virtual conference, which is titled as The Role of Polybasil Supplemented Diet for Growth and Immunity Promotion in Cygnus Mrigla Fingerlings. Fish is one of the crucial protein-rich staple food as it is highly digestible, rich in certain essential amino acids which are limited in terrestrial meat. Hence, there is continual rise in worldwide fish production. This increase in fish production causes intensification in the culture system. Intensification further causes overpopulation, which affect the health of fishes by poor physiological conditions and susceptibility to the diseases. To control and combat such infectious bacterial diseases, immunostimulants can be used. Synthetic immunostimulants may retain in the surrounding, therefore, there is drift in preference toward the use of herbal product as natural immunostimulants. Holy basil or Simum sanctum is known as queen of herbs as it is an extremely potent immunostimulant. Hence, present study was proposed to evaluate the optimum dietary incorporation level of holy basil or Simum sanctum for optimum growth performance and immune response of Cygnus mrigla fingerlings. Material method. For this study, experiment diet was prepared with the help of these 7 ingredients, and isonitrogenous isocaloric diet was prepared, which was given at the rate of 4% of the body weight of the fishes for a period of 90 days. This table represents the ingredient composition of the experimental diet in 5 treatment groups, in which amount of tussle leaf powder is 0 gram per kg in control, 25 in OS1, 30 in OS2, 35 in OS3, and 40 gram per kg in OS4. After that, fishes were procured and acclimatized in FRP tanks for 15 days prior to the experimentation. Then, 15 experimental tubs were set up. Each treatment group was in replica of 3. Each tub was stocked with 10 fingerlings and approximately 30 liters of water. 60 to 70% of water was renewed daily, and feed was given twice in a day for a period of 90 days. And length and weight of fishes was measured at every 15 days, and ration was adjusted according to it. Water quality parameter was also measured at every 15 days by following American Public Health Association 2005. After completion of the feeding trial, intestine was aspirated from the fishes, and it was determined the intestinal enzyme activity, such as amylase, protease, and cellulase activities was determined by following standard protocol. After that, hematological and immunological assessment was performed, in which TLC, TEC, phagocytic assay, NBT assay, serum protein, and turbidimetric assay was performed by following their standard protocols. Results. This figure represents the growth performance of Cygnus mrigla fed on various inclusion level of holy basil. It clearly represents that dietary treatment group OS3 fingerlings links show better growth performance as compared to other treatment groups. These are the polynomial fit curves fitting to the data of growth performance of Cygnus mrigla fed on various inclusion level of holy basil. The polynomial fit curve live weight gain, specific growth rate, and upend protein digestibility clearly indicate the optimum dose in dietary treatment group OS3. However, inverted polynomial curve was observed in feed conversion ratio with lowest FCR value in dietary treatment group OS2, which represents better utilization of feed in this group. Intestinal enzymatic, this figure represents the intestinal enzymatic activity of Cygnus mrigla fed on various inclusion level of holy basil. The dietary treatment group OS2 fingerlings show better amylase activity, better protease activity, and better cellulase activity. These are the box plot representing the effect of holy basil supplemented diet at varied inclusion level on total ammonia and total orthophosphate production. This box plot clearly indicates that dietary treatment group OS3 fingerlings show low ammonia and ortho production because of the better assimilation of the feed, low deamination, and hence low ammonia and orthophosphate production. These bar graphs represent the effect of dietary supplementation of holy basil at varied inclusion level on TLC, TEC, phagocytic ratio, and phagocytic index, representing the same peak in dietary treatment group OS3. This figure showing the phagocytic cells observed in Sinus migla after feeding trial of 90 days. These box plots representing the effect of holy basil supplemented diet at varied inclusion level on respiratory burst activity, serum protein, and lysosome activity, representing the same peak in dietary treatment group OS3. High growth rate was accessed in holy basil fried fingerlings as holy basil is a productive appetizer. Holy basil also increases the activity of enzymes present in the intestinal tract due to presence of high concentration of eugenol, hence increasing nutrient utilization. Holy basil is one of the competent natural immunostimulant as it contains water-soluble form phenolics compounds and a variety of other ingredients such as eugenol, methyl eugenol, and caryophyllene. Hence, it improves both specific and non-specific immune response. Hence, it is concluded from present study that inclusion of holy basil in formulated diet improved not only growth performance, but also hematological and non-specific immunological parameters in Sinus migla fingerlings. It is also evaluated that Sinus migla exhibit optimum growth performance and immunity at optimum incorporation level, and optimum incorporation level of herb holy basil is 35 grams per kg for sustainable culture of Sinus migla. The authors acknowledge the financial assistance provided by CSIR and New Delhi, India in the form of JRF. Thank you. Ma'am, I've finished my poster presentation. Am I audible? Ma'am, are you there?
Check. Check. Test. Check. Test. Check.
Dear participants, are you able to hear me?
Yes, ma'am.
Thank you, Deepika. It was a nice presentation. I have a question for you. You did you created a formulation, right? Diet.
Yes, ma'am.
And you took the. Did you take the whole plant or a part of the plant, holy basil, either the leaves or the seeds or something like that?
Ma'am, leaves. I took the leaves and made a powder. Yeah. Yes, ma'am.
Okay, the leaves. Maybe you did a literature search and through that you confirmed.
Yes, ma'am.
Okay. And what about you gave the formulation in the lab and it worked? Did you try it in the natural habitat where the fishes are grown or somewhere in the water?
Sir, ma'am, it was all under the laboratory conditions in the plastic tubs.
Yeah, you did it in the lab using a plastic bending, but
Yes,
when
ma'am.
it comes to a natural habitat, the fish will have many options. Here you fed only the formulation, but there the fish will have a lot of options, isn't it? It may or may not take the formulation. But is there any change with the natural water or the growing inside the lab and in the natural thing? Did you check that or you're yet to do that?
Ma'am, your voice is not.
Okay, that's fine, Deepika. Thank you for your presentation. We move on with the next presentation. May I request Jayalakshmi to present her work? Jayalakshmi, are you ready? Yes, Jayalakshmi from Rajalakshmi Engineering College, Chennai is now ready with her presentation. Yes, please proceed.
Yes. Is my screen visible, ma'am?
Yes, it is visible. Please carry on.
It's very good afternoon. Yes. Very good afternoon and all present here. This is Jayalakshmi doing MTech of second year from Rajalakshmi Engineering College. And I'm here to present my project on enhanced removal of cationic dye from textile effluent using adsorbent prepared from neem bark, that is Acidica indica bark. And I did my project under the guidance of Dr. K. Satya, Professor and Aid, and Dr. K. Nagarajan, Professor and Aid, Department of Biotechnology and Chemical Engineering. And firstly, introduction. Water pollution is caused due to discharge of effluents, textile and other industrial effluents into the water bodies. And this is considered to be the major pollutant which is found in the wastewater. There are certain techniques like flocculation, adsorption, oxidation, electrolysis, ion exchange method to treat this dye from the wastewater. Among these methods, adsorption is the one which is highly flexible in nature, it's cost efficient, and it's insensitive to toxic pollutant. And my scope of the study is to use this neem bark as adsorbent to treat this crystal violet dye from the industrial effluent. And firstly, materials and methods part. Here, first we have synthesized the activated carbon. First, the neem bark is taken and it is treated with acid in order to create a post to adsorb the dye. And after that, it is then treated with a muffle furnace in order to make a carbonized product. And the optimization studies was carried out, like varying the adsorbent dosage from 0.5 to 2 grams per liter. And contact time is between 20 to 100 minutes with a temperature of 25 to 55 parts per million. 25 to 55 degrees Celsius with initial dye concentration of 10 to 80 parts per million. And coming to the results and discussion part, here we have given SIM analysis to determine the surface properties of the dye after dye. And the first image shows the SIM image of untreated neem bark carbon, and the second image shows this crystal violet diluted neem bark carbon. And it is seen that the first image shows the rough surface morphology with the number of micropores to adsorb the dye, and the second image shows that the dye got adsorbed onto the surface and it is found to be fibrous in nature. And coming to the FTIR analysis, here it is found that the untreated neem bark carbon contains less number of peaks, whereas after adsorption, the peaks, the functional group that is present going to react with the dye and forms the complexes. And coming to the conclusion, here this biochar was prepared from the neem bark and it is used to decolorize the crystal violet dye from aqueous solution. And the following parameters were optimized, and the optimized adsorbent dosage is found to be 1 gram per liter, and with a contact time of 60 minutes, the temperature is 35 degrees Celsius and a dye concentration of 50 parts per million. And a same image shows the structural morphological changes before and after absorption of the dyes, and FTIR analysis shows the changes in a peak. And thus my results provide that this adsorbent has potential to absorb this crystal violet dye. And these are the references I have gone through to carry out my work. And thank you.
Thank you, Jayalakshmi, for the vivid presentation. I have a question for you. From where did you collect the textile effluent? Which part?
No, ma'am. Initially, we prepared, ma'am, collecting the dye.
Okay, so it was your own preparation, just to.
Yes, collecting the dye from a market as a powder, and we prepare, ma'am.
Okay, so how did you get the idea of using neem bark? Why neem bark in particular? Is that
actually first thing is yes, ma'am. It is a byproduct waste, ma'am, and it is seen that no work has carried out using this neem bark to treat any kind of dye, ma'am. And it's like converting waste to wealth.
Okay, okay. So thank you, Jayalakshmi, for the presentation. We move on to the next presentation. May I request Seena KK to present her work? She's from St. Thomas College, Thrissur. Yes, Seena, can you please raise your hand so that we can unmute you? Seena KK, kindly raise your hand. Ms. Seena, we have unmuted you. You can now present your poster.
Okay, ma'am. Am I audible now, ma'am?
Yes, yes, we can hear you. Please go
Can
ahead.
you see the screen also?
No, please try again.
Okay, okay, ma'am. Hello, ma'am. Can you see that screen?
Yes, I can see your poster.
Okay. Good morning, all. I'm Seena, research scholar, Department of Botany, St. Thomas College, Thrissur. And my topic is on a study on the diversity of euglenoid species and evaluate the water quality of Bharatapuzha River Basin, Kerala. About introduction, as we know, in the importance of freshwater ecosystems and the study of phytoplankton, especially in the case of biomonitoring analysis, because most of the phytoplankton, especially euglenoids, they have the ability to pollution tolerant. That is, they have tolerate pollution. That's why they have used for the indicators of organic pollution. In my study area, I have selected the Bharatapuzha River Basin, that is second longest river in Kerala and a major river in Kerala district. From Bharatapuzha River Basin, I have selected 10 sampling stations. From these 10 stations, I have selected the samples for both physicochemical parameter analysis and also phytoplankton analysis. In methodology, I have selected 2 liter samples for the analysis of both the physicochemical parameters and phytoplankton. And in the case of physicochemical parameter analysis, I take mainly I have selected 6 parameters, that is temperature, pH, electrical conductivity, nitrate, phosphate, and silicate. At the analysis for phytoplankton, I have selected 1 liter sample, and this is preserved with the 4% formalin solution. And this sample concentrated to 10 ml after centrifugation. And this sample used for the analysis of phytoplankton. And then photomicrographs taken for the analysis of analysis and identification of phytoplankton. About result and discussion. While analyzing physicochemical parameters, both temperature and pH were found to be high during the present study, and also that is high at the time of pre-monsoon season. The remaining parameters, like especially nutrients, they were within permissible limit. The major increase seen in the case of both the temperature and the pH. And at that, while analyzing phytoplankton, totally I got 59 taxa from the study area. That is that belongs to 5 genus. That is Euglena, Tracheomonas, Leposinglis, Phacus, and Strombomonas. These are the 5 euglenoid genus obtained from the study. And among these, the most dominant genus, that is Euglena. Then this graph shows the maximum number of average number of cells per liter collected from different months. These months like March, April, and May, that is especially in pre-monsoon season, have got a max.
Sina, I'm unable to hear you. Is your presentation over? Yes, Miss Sina. It's not audible. Can you please check your internet connection? Sina, are you able to hear me? Yes, I think there's some technical problem from Sina's side, but that's all right. We shall move to the next presentation. May I request Pooja Tak to present her screen? Pooja, are you ready with your presentation? Ms. Pooja, are you ready with your presentation?
Yes, ma'am.
Yes, please carry on.
Ma'am, my screen is synced. Can you hear me?
Yes, we can hear you.
Okay.
Yes, please proceed.
Yes. Good afternoon to all. Myself Pooja from General University, Jodhpur. Thank you for giving me a great opportunity to present here my research work. My topic is a kinetic and mechanistic studies in the oxidation of substituted benzaldehyde by pyridinium dichromate. First of all, we do in this research, we choose a oxidant which is very suitable for according to green synthesis. Then chromium salts have been used as oxidizing agent in synthetic organic chemistry. Because of that, they are very drastic compounds of this and easily soluble in all solvent. So we used chromium salts, and chromium-6 have been prepared and used in organic synthesis as mild and selective oxidant in non-aqueous solvent. And for this, we formed a pyridinium dichromate is also one of the such compound, and oxidation of some monosubstituted benzaldehyde by PDC in DMSO as solvent. In this, we first of all form an oxidant like pyridinium dichromate. Then we use it as a substrate benzaldehyde in as a solvent DMSO. Some structure is here, and this picture is for DMSO PDSC crystal. The major objective of this investigation was to study the structure reactivity correlation for the substrate undergoing oxidation. In reaction studies and methodology, first of all, we formed oxidant, and after that substrate and also solvent is used as a DMSO. Study the correlation analysis of reactivity in the oxidation of substituted benzaldehyde by pyridinium dichromate. We proceed a reaction. After that, we obtained a reaction, which is a first order reaction, in which we see that both reaction acid dependent path reaction and also acid independent path. In this aromatic aromatic benzaldehyde react with the PDSC, pyridinium dichromate, and formed a carbonyl compound. Then after that, second path in which acid dependent path, the path, my voice is not hearing. We sorry to say.
We can hear you. Please don't worry. Carry on, please.
Sorry, actually. Okay. After that reaction proceeding, we collect some data from the instrument. We use a spectrophotometer, and we observe for oxidant as a lambda max. And after fixing the lambda max, we reaction proceed like we used M by 1000 pyridinium dichromate and aldehyde and TSO2, toluene sulfonic para-toluene sulfonic acid as used as a catalyst. Because of that, without catalyst reaction is very slow. So we observe that after that we reaction proceed different concentration. We observe K values, all of that K values, and this reaction is significance of study. It helps to optimize the reaction condition for synthetic studies, choice of catalyst, solvent, nature of substituent like pH, also temperature, etc. And quantitatively structure reactivity analysis is highly useful in many fields like medicinal chemistry. Because of that, according to this procedure, we obtain a rate of reaction, and also we study a rate laws like first order, pseudo order, second order reaction, which help in the reactivity analysis. And kinetic studies are essential in evaluation of efficiency of catalyst, photochemical reaction, and in environmental study. We have some future plan like pharmaceutical in photosynthesis, also in green synthesis, that we use all of rate of reaction that we used how to proceed, how to effect of temperature, that is. And after all that, conclusion is that the reaction is proposed to proceed through a hydride ion transfer. It has also been observed that an alpha carbon hydrogen bond is cleaved in the rate determining step. And we observe that both protonated and deprotonated forms of PDC are the reactive, reactive oxidizing species. And initially chromium 6 is reduced to chromium 4. It is likely to react with another chromium 6 to generate chromium 5, which is then reduced in a fast step to ultimate product chromium 3rd. Such as a sequence of the reaction is oxidation is well known. Thank you.
Thank you, Pooja, for your presentation. You have substituted benzaldehyde. Did you try with the other aldehyde groups, or what is the reason you have specifically chosen benzaldehyde?
Yes, ma'am. Ma'am, your voice is not clear. Can you repeat the question?
No, I just asked, is there any specific reason you chose benzaldehyde? Did you try with other aldehydes or something like that?
No, ma'am, actually we proceed further further aldehydes like benzaldehyde, formaldehyde, acetaldehyde, but we see that the in benzaldehyde reaction is very convenient other than aldehydes.
Okay. So thank you, Pooja, for your presentation. We shall move on to the next presentation. Shall I invite M. Ramya to present her work? Ramya, could you please raise your hand? Yeah, okay, yeah, right. Just a minute, please wait. Yes, Ramya, you can share your slide now.
Good morning, ma'am.
Yes, good morning. Please go ahead.
Am I audible?
Yes, you are audible, Ramya. You can proceed.
Good morning, one and all present here, and thanks for the great opportunity to present my topic, cyanobacterial identification of deacetyl using 16S DNA-based molecular method. I am working in, I'm from Presidency College, Chennai. My guide is Dr. Tari Uma Maheshwari ma'am, and the co-guide is Sanyasi Erumalai sir from Madras University, Chennai. I have collected, the lakes are, we all know the lakes are one of the most biological drivers in the productive inland surface of water habitats. Why I have chosen this lake especially because in Chennai, whenever the Chennai is facing the drought, so in the summer season, we will get the drinking source from the Kadalur district Viranam lake water only. This, this lake water, the Viranam Lake is actually, earlier it was called as Viranarayapuram Lake, located in 14 kilometers.
Sorry to interrupt you. We are unable to see your poster. Can you please share your poster and then continue with your explanation?
Oh, sorry, ma'am. Is this, can you see the screen, ma'am?
Yes, now it is visible. Please carry on.
Okay, sorry for that. The lake I have chosen is the Viranam Lake, which is located in the Kadalur district of Tamil Nadu, India. This lake capacity is 1,465 million cubic feet, and it is 14 kilometer, which is quite very big capacity of the lake. This earlier it was called as a Viranarayapuram Lake. which was created by the Rajaditya Chola, named after the Parantaka Chola, who was his father. He was, because the Parantaka Chola got the title as the Veera Narayanan. So the lake has been named after him. Why I have chosen this lake is because whenever the Chennai is facing the drought and during the summer season, we will get the lake water from this Viranam lake only. We are so curious to know about the parameters, the physicochemical parameters of the lake. When I have analyzed the 24 different parameters of including the heavy metals, I found that there was a the great biological oxygen demand was there. So because of that, the fishes were exposed to the toxic of cyanobacteria. The cyanobacteria was isolated from the Viranam lake water. This is the place where, here the lake is actually located in Tamil Nadu. So actually, initially when I have isolated and grown in the culture of BG11 media, I have found out 10 to 15 of contaminated cultures, a variety of cyanobacteria and macroalgae, microalgae, everything was there. So to get the axonic culture, I have serially diluted and then was plated, and the agar plate was carried out, and I have isolated the single colony axonic culture. Sometimes I've used the imipenem antibiotic as well to get rid of some bacteria. Since it is a cyanobacteria, it's a prokaryote, but it acts as an eukaryote. It just gets contamination. Definitely from the bacterial contamination would be there, because the glycoprotein which is surrounded by the algae will get attracted, the bacteria will get attracted to the sugar present in it. To get rid of that bacteria was a difficult task for me, and 3 of the culture I have isolated, and 1 I have got the novelty work, which was I got the good results but not matched with the NCBI. So I have yet to put the name and get the patent. These are all the 2, I mean, this is one among the culture, and this is 40x and this is 100x image of the decitabile cyanins. This DNA was isolated from the culture fragment of 16S rDNA gene, was amplified by using the forward and reverse primer, which was purified later on and to remove the contaminants. The forward and reverse DNA sequencing reaction of the PCR amplicon was carried out using the BDT V31 cycle sequencing kit on AB1370X genetic analyzer. The methodology for the isolation of DNA, I have used the CTAB method, and for the phylogenetic analysis, the evolutionary history was inferred by using the maximum likelihood method based on the Chimera 2 parameter model. The bootstrap consensus tree inferred from 1000 replicates, which is taken to represent the evolutionary history of the taxa analyzed. Branches corresponding to the partitions reproduced in less than 50% bootstrap replicates or collapsed. The percentage of replicates trees in which associated taxa clustered together in the bootstrap test, which was 1000 replicates, are shown. So the initial trees for the heuristic search was obtained automatically by applying neighbor join and BioMG algorithms to a matrix of pairwise distance estimated using the maximum composition likelihood approach, and then selecting the topology with a superior log likelihood value. The codon position included here was the first, second, and third, and including the non-coding region. All positions contained the gaps and the missing data were eliminated. These were the total of 1434 positions in the final data set. Evolutionary analysis were conducted using the MEGA software. Here I conclude the results and discussion said that the discrete PCR amplicon band of 1500 base pairs was observed using the 1% agarose gel. Using the BioEdit software, I have just put the sequencing forward and made it a complementary sequence, and the consensus sequence of 16S rDNA was generated. And when I blasted it in the NCBI database GenBank, the first maximum identity score of the first 10 sequences were selected and aligned using the multiple software program, Clusterla, and the distance matrix was generated and the phylogenetic tree was constructed using the MEGA 7 software. Here I conclude that the aquatic habitat of the new material from Viranam Lake revealed a wider distribution of members of the genus. The new taxon was found to be the closest to Desertiphyllum parens. which is oscillatorials. It's a separation of the new species as Disciphyllum xylens using the conventional the molecular traits. So the feature of the organism is Disciphyllum xylens. The reports in the bacteria of cyanobacteria, Oscillariophysidae, oscillatorials, and the Disciphyllum. So these are all the bands that I have observed. This was the 1500 base pairs in the agarose gel. Here I conclude that my aim is to work for the toxin study based on for the hepatotoxin study, and my research work is still going on. Isolated the cyanobacteria was extracted using the ethanol extraction and then given for the LCMS in the IIT Bombay. And using the fish analysis and the ELISA kit, the toxin was found out. And this is one of the top work in my research. I hereby acknowledge that thanks Dr. Professor Erumalisa for supporting me for this research work to do in his laboratory. Thank you.
Thank you, Ramya, for your presentation. So
Thank
you took the water samples, and which media you used to grow these cyanobacteria, because you said
it is a BG11 media. I have standardized the BG11 media, and initially the pH, I was changing the pH with the 7.4, 7.3, 7.1, and in that 7.1 pH, I have got this particular cyanobacteria, and some in 7.3 pH of cyanobacteria. And some was growing for 6 months, was no growth was found out. So after 6 months, the cyanobacteria started growing. So these are all the pH I have changed, and the BG11. This is a blue-green algae of 11 medium.
Okay, because it's very difficult to grow algae.
Yes, ma'am. Yes, ma'am. Also, the sequencing result which I have got, I have submitted in the NCBI. With my accession number is OM349562.1. So this initially carried out the sequencing was very difficult for cyanobacteria, because most of the cyanobacteria was not mentioned in a database. So all the time I'll get the sequencing results, but the phylogenetic analysis was not able to follow up. And so this is how I've got the result. Thank you.
Okay. Thank you. All the best for your work. So we move on to the next presentation by Tara Paul. Tara Paul, can you please raise your hand? We shall move on with the next presentation. May I request Tara Paul to do the next presentation? Tara, please share your screen.
Ma'am, is it visible?
Yes, Tara, please carry on.
Good afternoon to all. Myself, Tara Paul, research scholar from Christ College, Irnyalakuda. The topic of the study is distribution and identification of bacteria from mangrove sediments of North Kerala. A fisherman from Thailand said that if there are no mangrove forests, then the sea will have no meaning. It is like having a tree with no roots, for the mangroves are the root of the sea. So it is the importance of mangroves. The ecosystem is characterized by periodic tidal flooding, which makes environmental factors such as salinity and nutrient availability highly variable, resulting in unique and specific characteristics. Mangroves provides essential habitat for thousands of species. Apart from flora and fauna, microbial diversity characterizes one of the important communities of these ecosystems. In tropical mangroves, bacteria and fungi constitute 91% of the total microbial biomass, whereas algae and protozoa represent only 7% and 2% respectively. Complex interactions of these microbes maintain the harmony of different biogeochemical processes and sustain the nutritional status and ecological balance. The mangrove environment is a rich natural resource that is currently underused. Microorganisms submerged in the depth of mangrove sediment are one of them. Therefore, this study aimed to isolate and characterize mangrove sediment bacteria from northern Kerala. Coming to materials and methods, mangrove sediment sampling. During the 3 seasons, that is monsoon, post-monsoon, and pre-monsoon, mangrove sediment samples were collected from 8 different locations in North Kerala. It is depicted in figure 1. The subsurface mangrove sediment sample collected using hand core collection method and transferred aseptically in the polythene bags. Then isolation and purification of bacteria. The sediment sample was serially diluted, mixed well, and performed spectral technique and incubated overnight. Purification was carried out by separating the morphologically different bacterial colonies in each petri dish based on their characteristics. Coming to characterization, it is based on observation of microscopic morphological characters using gram staining and physiological characters using biochemical tests such as the TSI, oxidase, catalase, indole, MRVP, citrate, urease, etc. The results were analyzed and interpreted using Burgess' Manual of Systematic Bacteriology. Then result. Present study obtained 526 isolates belonging to 31 different bacterial genera that were successfully purified, which proves that mangrove ecosystems are rich in bacteria. The result obtained depicted in figure 2 and 3. From the pie diagram visible that the obtained genera from the sample are Bacillus, Enterococcus, Xanthobacter, Klebsiella, Lysobacter, Pseudomonas, Staphylococcus, Serratia, and Vibrio. The remaining 22 genera represented in others because of less percentage obtained. It's about 1 to 2%. The line graph shows the bacterial variation in different seasons. Bacillus species present at high percentage. Bacillus, Xanthobacter, and Enterococcus are the predominant genera among bacterial isolates. From the isolates, 54% were gram positive and 46% were gram negative. Several previous studies were supposed the findings. The prevalence of bacteria didn't follow in correct pattern. The percentage varies according to species and seasons. The number of bacterial isolates is high in monsoon than other seasons. Bacillus species has industrial applications used in the production of enzymes, amino acids, antibiotics, vitamins, etc. Likewise, several studies shows that Xanthobacter and Enterococcus used for bioremediation purposes. The other isolated bacteria are belonging to the category of nitrogen fixers, phosphate solubilizers, bioremediators, sulfur oxidizers, etc. Then coming to conclusion. Mangrove sediments were rich with bacteria. The data about the composition of bacteria provides better understanding about the mangrove habitat and process take place. The bacteria in the ecosystem is unique because of the adaptation occurred from such versatile environment. Hence, the proper utilization of potential bacteria are promising future for human life betterment. Now I acknowledge KSST for financial support for the study. Thank you all.
Thank you, Tara, for your presentation.
Okay, ma'am.
So you isolated the bacteria from the mangrove area. Is there any category of of bacteria that was predominant? Like you said that sulfur oxidizers, you isolated different varied
yeah, ma'am. I isolated the nitrogen fixers, that's the Bacillus, Xanthobacter, Enterococcus, and others, the Acinetobacter, Serratia, are reported to be nitrogen fixers.
Okay, which category was predominant in that particular area? Which?
It is the Bacillus species.
Priceless. Okay, so you checked it in the lab. You took samples from the mangrove area and you did it in the lab. Is there any difference between the growth of the bacteria inside the lab and in the natural habitat? Do they show any difference? Are you able to successfully grow all types of microorganisms, or some microbes grew very okay in mangrove area?
Yeah, it's the in vitro conditions mainly. Except some actinomycetes and all, in the laboratory condition, the majority of bacteria, some of the bacteria could not grow. That's why we ongoing some work on NGS.
Okay, so thank you, Tara. Next, we move on to the next presentation by Shruti Yalani. Shruti Yalani. Shruti Yalani. Now you can share your screen. Please unmute.
Yes, ma'am.
Yes, please carry on.
Ma'am, my screen is visible, ma'am.
Yes, yes, please carry on.
Okay. Good afternoon, everyone. I'm going to talk about textile dye decolorizations of textile dye decolorizations using bacterial strains. So first introductions, dye decolorization means the process of removing colors from the waste or effluents by means of some biological or physiochemical mechanisms like bioaccumulation, bioabsorption, and biodegradation. So in other words, it means the large dye molecules are broken down into smaller molecules by chemically. So by the end of this, we have the byproducts like water, carbon dioxide, and mineral byproducts. Then my present study objectives includes isolations and screening for the particular reactive red textile dye decolorizing bacteria, and to select the potential isolate for reactive red dye decolorization, and to optimize various parameters, and to study the role of enzyme in dye decolorizations and biodegradations activity was performed by using UV spectroscopy analysis, and to study the effect of immobilized bacterial cells on dye decolorizations. At last, toxicity analysis was performed. My methodology include isolations of dye decolorizing organisms, decolorizations experiments and assay, various parameters with checks like incubation time, temperature, pH, initial inoculum, effect of initial dye concentrations. And similarly, I performed the enzyme characterizations and biodegradation studies and immobilization studies. At last, I tried with some cheap fruit pasters like sapota, custard apple, and apple to decolorizing. Finally, molecular identification was performed to determine the potential isolate. These are my results. I collected totally 11 soil samples from Chennai, and here 8 bacterial isolates were found to show clear zone around the colony for reactive red decolorization by plate assay method. Among the 8 isolates, I found 1 potential isolate was selected based on the maximum decolorization by the broth assay method. Several parameters I already tell, the incubation period, temperatures was performed. So, and maximum decolorization was performed 18 hours at 32 degrees Celsius. In case of pH, the maximum decolorization was observed and maintained at 7. Inoculum concentration of 1% and the dye concentration is 0.1 mg per ml was found to be optimum condition for this reactive red decolorization. And for the immobilization study, the azo reductase activities was in the reactive red decolorization broth was determined and found to be 12.52 U ml minus in. The partially purified fluid extract was characterized by using the SDS page and distinct bands were observed. And finally, I determined the biodegradation analysis for the decolorized product by using the UV visible spectroscopic method. Immobilized bacterial on the reactive red decolorizations was studied by using the sodium alginate. Finally, I used 3 different fruits like custard apple, apples, and sapota. Among these 3, the custard apple shows the more amount of decolorizations. In microbial toxicity, I performed the well diffusions method, and phytotoxicity seed germinations method was performed. I took 3 types of seeds, cowpea, green pea, and another type chickpea. These 3 types of seeds were taken and performed with various distilled water, reactive red, and nutrient broth. And among 3 seeds were decolorized. And finally, the potential isolate was identified as Bacillus cereus by using the 16S rRNA DNA sequencing. The gene sequence of Bacillus cereus strain BIOS MD2 was deposited under the GenBank with accessions number. So finally, I concluded my topic. I finally level soil samples from that 1 potential isolate was selected and screen by 16S rRNA DNA sequencing, and the GenBank with accession number was founded. So comparatively, what my suggestion is, compared to another artificial techniques, biological decolorization is the most effective and eco-friendly approach than the physical and chemical method. Thank you, ma'am.
Thank you, Ms. Shruti Alani, for your presentation. So you use textile dye.
Yes, ma'am.
From where did you collect the soil samples to isolate the different bacterial strains?
From Tirupur, ma'am.
Tirupur, what type of soil you used? Is it a normal soil or you took it from a particular area?
Like around the industry, ma'am, around the dyeing industry, I took soils, and after that purification process, some sludge samples were collected, ma'am.
Okay, okay. So you took it near the textile dye.
Yes, ma'am.
Okay. What textile factory where you collected the dye? Is it a what kind of factory is that? Textile
Dyeing industry, ma'am.
Dyeing industry. Okay.
Yes, ma'am.
Okay. So did you try that with the fungal strains? Because usually they say fungus is more effective.
Yes, ma'am. But I already did this project, ma'am. I continued with the white rot fungi, ma'am. I focused on this, ma'am.
Okay. So thank you, Shruti Alani, for your presentation. May I request Uma Maheshwari to present her work?
Thank you, ma'am.
Dear participants, this is the final call for the Group 1 poster presentation members. Shweta Kashyap, Anju Kumari, Murugash, Uma Maheshwari, and Kumar Amit. So if you're ready with your presentation, you can raise your hand. This is one final call for the participants of Group 1. Yes, thank you very much, dear participants. From the Department of Biotechnology and Bioinformatics, we wish you the very best for your future research work. Keep researching and please be in association with us. Thank you so much for your active participation and all the very best to everyone. We now break this poster presentation session. So after lunch, we shall assemble again So I request all the participants to join at 1:30 p.m. afternoon for the post-lunch session. May I repeat? Kindly join at 1:30 p.m. for the post-lunch session and the validation function. Thank you.
Good morning, ma'am. Good afternoon. Ma'am, can I start my presentation now? Ma'am, am I audible, ma'am?
Yes, sir. Uma Maheshwari, you can share your screen.
Yes, ma'am. Thank you.
Yes, Uma Maheshwari, you
Yes,
can.
ma'am, I'm sharing.
Yeah, okay.
Post disabled participant screen sharing in the portal. Ma'am?
Yes, I
Post is
can hear
disabled.
you, but the slide is not visible.
No, ma'am. It has post disabled participant screen sharing. Post is disabled, ma'am. Shall I tell you the PPT, ma'am? Slide?
I'm not able to hear you, Uma Maheshwari.
Am I audible now?
Yes, you are audible, but I'm not able to see your screen. Please share it once again. Please try again.
Post disabled participant screen sharing.
Please wait for a minute, Uma Maheshwari. Yes, please try now. Can you please try now?
Yes, ma'am. Is it visible now, ma'am?
Yes, I can see your screen. Please continue.
Yes, ma'am. My topic is on a review paper, which is on a review on the medicinal properties of Sorry. First, I introduce myself. I'm Uma Maheshwari, doing my research work in biotechnology in Srimad Andavan Arts and Science College. And my guide is Dr. B. Chitra, ma'am. Ma'am, my review, I'm going to present a review paper on the plant Centrathecum punctatum. Am I audible, ma'am?
Yes, you are audible. Please continue.
Yes, ma'am. This plant, Centrathecum punctatum plant, is a plant which belongs to a Asperis family, which is very much valued as a medicinal plant in the olden days. From traditional period itself, it has been used for many for solving many ailments like for inflammation or tumor depression or any anti-proliferative purposes, ailments. And recently, for that, in those days, they don't know how to, what is the concept behind this. But nowadays, it has been found from this, from reading this plant. I have come to know about the medicinal properties of this plant. It is very rich in its bioactive molecules, like it is having anti-cancer property, antimicrobial, anti-proliferative property, as well as anti-hypertension relieving agent, anti-aging property, anti-lipido agent, lipidomic agent, and it is used as a hair preparation oil. The common name of this plant is Kesavardhini. So usually people will be using this plant for hair oil preparation, and other than that, it has been used for skin whitening, anti-aging, and it is used as a painkiller, antidote for snake bite and as well as tiger bite. And this plant, it has been a very active biocomponents, secondary metabolites in it, as it is used as an anti-hyperglycemic activity. And this plant has more cytotoxic activity, and it is used as an antioxidant. It is having an antioxidant property. And the previous studies of this plant are in vitro cytotoxic antioxidant and GCMS studies on this aerial parts of this plant shows that many components, bioactive components in this plant is having antioxidant, anti-cancer property, as well as the all the alien parts are used as a cytotoxic anti-cancer agent. And other than this, the leaf extracts of this plant is used for antimicrobial, antioxidant, and anti-proliferative property. Then phytochemical screening of this plant, phytochemicals, as well as GCMS analysis of this plant shows that it is having more activity. It is evaluated that it has more anti-inflammatory activity. Of all these studies from this review, what I found the result is the methanol extract of this plant, whatever it may be a flower. If a flower extract is taken or a leaf extract is taken, it is very active and it shows more activity than compared with the other solvent extracts, like using petroleum ether or ethanol extracts. This methanol extract of this plant is very effective in showing all these medicinal properties. And I conclude that this plant, as from this review, what I conclude is this plant is having more medicinal property. Instead of using the allopathic treatment, this can be used as an alternative for the various ailments which are we are facing in day-to-day life. Thank you, ma'am. Thank you for your patience.
Thank you, Uma Mangeshwari, for your presentation. You can leave. Thank you.
Thank you so much, ma'am. Thank you for giving me an opportunity, ma'am.
Thank you. You're most welcome.
Hello. Hello, guys. Can you hear me?
Yes, sir.
Oh, very nice. Can I start my talk?
So wait a minute, sir.
Okay.
The afternoon session is about to begin. I invite Dr. Suvaithen Amudan, Assistant Professor, Department of Biotechnology and Bioinformatics, to introduce the respected speaker, Dr. Ravi Manjithaya, Associate Professor, Molecular Biology and Genetics Unit, Jawaharlal Nehru Center for Advanced Scientific Research, Bangalore.
Good afternoon to all. Dr. Ravi Manjut has the degree of the in postgraduate Indian Institute of Science in Bangalore. He did the postdoc in the related to Indian Institute of Science and the University of the California in 2011. In the Ravi is joined in the Molecular Biology and Genetic Unit in Jawaharlal Nehru Center of the Advanced Science Research Faculty of the following the current working as the Associate Professor and Associate Professor in the Neuroscience Unit. Dr. Ravi has published the 36 research paper, 12 review papers in scientific journal of the citation in the 10,249 citation, H index in 16. I attendance in 30 is the 2 award is received in the Dr. Ravi, the Center Drug Research Institute of Award of the year of 2020. The another award is the test in the DBT in the fellowship of the 2011 to 2016. The invited, the honorable speaker of the Professor Ravi Manjut, the individual lectures. Thank you, sir.
So you can proceed, sir.
All right. Let me just share my screen. Can you see my screen?
Yes, sir, it's visible.
Can you see the full screen also?
Yes, sir.
Okay. So I'll just introduce myself. My name is Ravi Manjithaya. I'm an associate professor studying cell biology, in particular autophagy, at Bangalore. This is a central government research institute called as Jawaharlal Nehru Center for Advanced Scientific Research, and in the Department of Molecular Biology and Genetics Unit. I'm also associated with the Neuroscience Unit in this institute. I did my education from Indian Institute of Science, where I got my PhD. And then I went to University of California at San Diego for to get my postdoctoral training. And since then, I've been working here at CSIR in trying to understand how autophagy happens in all our cells and what can we learn from it, especially how it contributes to keeping us healthy and what can we do to manipulate it in certain diseases where it can be beneficial. Okay, so therefore, my title of my talk is the role of autophagy in health and disease. As you can see here in this panel, there are yeast cells, there are human cells, and there are also plant cells. Okay, all this work is from our lab, and what you can see that in yeast cells, for example, individual round, these are individual yeast cells. These yeast cells are used to make beer, or they're also used to make bread, for instance, and they're known as Saccharomyces cerevisiae. These are human cells. The most common ones that are used in research are known as HeLa cells, for instance. And then you have plant cells here, which is a fancy plant cell known as Aponogeton madagarensis. And in all these cells, you can see there are some dot-like structures that are here. Here also you can see many colorful dot-like structures. And here in the plant cell also, which is the boundaries are here, the cell wall, you can see dot-like structures going inside. So what are these dot-like structures and why are they so important? So if you study them, and this is a cartoon trying to explain what these dot-like structures are. And one of the things about eukaryotic cells is that eukaryotic cells have this amazing ability to form a Pac-Man-like structure. For old school gamers on computers, you must have heard of a game called Pac-Man. And in this Pac-Man game, you can see the cells can make a double membrane vesicle, which can engulf portions of the cytoplasm and gobble up anything like proteins, here in mitochondria as well, and pack them like this. The advantage of doing this is that whatever it has captured inside our cytoplasm is now sequestered from the main cytoplasm. And if it is any damaging causing stuff inside our cells, which has been captured in this double membrane vesicle, when it fuses with another organelle known as lysosomes, many of you must have heard of lysosomes as suicide bags inside a cell because they contain a lot of hydrolytic enzymes which can break down. It's more like a mixer inside your kitchen that you have. And when it fuses with this double membrane vesicle, it chops off everything. All organelles, proteins, everything gets degraded. And what gets thrown out into the cytoplasm are these building blocks known as amino acids. So if you break down proteins, you get amino acids. If you break down nucleic acids, you get simple nucleotides. And then complex fatty acids and sugars can be broken down to simple fatty acid components and sugars. All of that is pumped back into the cytoplasm. So not only autophagy collects all the garbage from the cells, but it also recycles in essential building blocks to the cells. So it serves two purposes. And this recycling function is very, very important because when there is not enough food outside, then the cells will rely on autophagy to recycle its own material and provide food and nourishment to the cell for survival. Okay, so autophagy is very, very important during starvation response and also to keep your house clean. All right. So I just want to take a quick check here. Are my slides fully visible and can you clearly hear me?
Yes, sir, it's visible, sir.
Very good. So I'll continue then. Because I'm unable to see you guys, I'm not sure whether I'm clearly visible, slides are clearly visible and able to hear that. Okay, so how does this process work? And so here in the process of autophagy, you can see that inside our cells, there are these mitochondria and there are these peroxisomes and many other proteins and other cellular material like ribosomes are there. And these are then captured during autophagy by a double membrane vesicle, which begins as a phagophore to expand and form this double membrane vesicle known as autophagosomes. And so autophagosomes are the real business end of autophagy. When they are formed, they can choose to capture anything. And here they can go and fuse with lysosomes, or in the East, they are called as vacuoles, which contain hydrolytic enzymes that will then degrade everything inside it. And so you can not only have capture and clearance, but also recycling materials. Okay. And so this was first observed by Professor Christian de Duve. He got a Nobel Prize in Physiology and Medicine in 1974 for actually discovering lysosomes and function. But when you're studying lysosomes, he did observe at that time autophagosome-like structures, and which he coined the term autophagy. Autophagy means self-eating. So cell eats part of its own stuff for recycling and for energy purposes, and also to remove the garbage. Okay, and so he got the Nobel Prize for lysosomes, but he did coin the term autophagy. But how autophagy works? What are the proteins involved in autophagy? What regulates autophagy? All of that remained quite a bit of mystery till Professor Oshumi did his famous screen, for which he got a 2016 Nobel Prize for discovering the mechanisms for autophagy. Okay, and he's a very inspirational figure. You can read about him, and his talks are there on YouTube also. And his success mantra was that he always wanted to work on something that nobody else worked on. And so although autophagy was known, but nobody knew about mechanisms for autophagy, and that's what got him going. Okay, and he also has a very nice point to make, that he says he always likes to get to the bottom of the problem, which means that if you guys are getting stuck because in some problem, you should not let go of it, but you should continue till you get solve the whole problem. And that's what he was trying to say. Okay, And he also has a message in his Nobel Prize talk for young people. And he says, I'd like to tell young people that not all can be successful in science, but it is important to rise to the challenge. So we should always not measure everything, whether we are successful or not, but it's important that whether we can give our 100% to any problems that we may be interested in. Okay. And so this is also the motto of doing research. Is research can be extremely difficult, but it's extremely rewarding also, so long as one is persistent in addressing all the problems and systematically we try to solve those problems. So I told you about this process of autophagy, and here in the cartoon we are seeing the same thing. A double membrane vesicle can come and capture cytoplasmic contents, including protein and organelles. It completes and forms this double membrane vesicle like an autophagosome, and then it fuses with the lysosome for forming an autolysosome, which will degrade everything inside and throw out recycled products such as basic amino acids, nucleotides, sugars, and lipids. Okay. While now we know some of these things in autophagy, what is most interesting aspects of one of the most interesting autophagy aspect is how is this double membrane vesicle formed? What controls? How many such vesicles can be formed inside a cell? How fast can this process happen? How much can we degrade? How much can we recycle? And these are the questions that are still a bit open in the field. A lot of people are addressing it. In fact, one of the review comments were also that everybody is trying to find this autophagometer, meaning by is there a way or a mechanism by which we can control this process? Either you can make it go faster, go slower, make it go more, make it go less. And so that is also what our lab is interested in. And I will be today telling you about 3 such stories from my lab, trying to address how autophagy is regulated. First one is we study yeast, and using yeast model, we address the basic mechanisms of autophagy. The amazing thing about autophagy is that it is conserved, meaning by autophagy happens in yeast cells, in plant cells, as I told you, in mammalian cells and everywhere. And so we use these models to study how autophagy or autophagosome biogenesis happens in yeast. While autophagy can devour a lot of stuff, it can also devour our intracellular pathogens. Any kind of infection that enters our cells can also be captured and cleared by autophagy. So it's an adaptation or a beta version of autophagy, if you wish, which can now be suited to clear such pathogens that enter our cells. Alternatively, autophagy can also be used to clear protein aggregates, which are causative of many neurodegenerative diseases, such as Alzheimer's and Parkinson's. So I'll tell you these 3 stories, and some of the publications that we've gotten from the lab is put here, okay? And so here is a story from yeast, and what my students Sunena and Ruchika now are doing is to use yeast and use fluorescence microscopy. So this is one yeast cell, and here is the target. This pink colored stuff is something that the autophagy wants to capture and gobble. So what I'm showing you here is that how this green colored autophagosomes goes around this blue colored cargo and try to capture it and eat it. Okay. And so that is an interesting part. So by studying these and doing something known as genetics and combining it with biochemical mechanisms, we know that what are the proteins that could be playing a role in this. If there is no autophagosome biogenesis, then this cargo will not be captured. But by doing this kind of studies and asking which protein is important, and if that protein is important, then when you delete that protein or knock out that protein, right? So in the absence of that protein, this process will not happen. And by doing that kind of studies, we identify new proteins that may be involved in autophagosome biogenesis. Okay, and so you can study and image such events inside the cells that contribute to clearance of cargoes. We also want to study how biogenesis happens. And so this is the factory inside a cell which makes autophagosomes is known as PAS, P-A-S, pre-autophagosomal structure or phagophore assembly site. Okay, and in these sites, what happens is that autophagosomes are made, and we don't know how exactly they are made, although we know there are many proteins that participate in this process at various stages, but exactly how it is built, we don't have a complete idea. And in our studies, in our lab, my students, Gaurav, he also identified and characterized proteins called septins. These form ring-like structures around this growing, budding autophagosome. Okay, and these ring-like structures shown in green here, we think are important because they form like a scaffold. You know, when you blow out soap bubbles, you must have seen a ring-like structure that is there, and then you have to dip it in soap solution and then blow out a soapy bubble, and that's how it happens. And in a similar fashion, we think septins may also be contributing. So by using the power of yeast genetics and fluorescence microscopy and combination of cell biology techniques, one can identify new proteins that are involved in this process and try to get a better understanding of how autophagy happens inside a cell. Right now, we are working on this to characterize whether this process happens in further or not. Okay. The other advantage of using yeast is that you can have very interesting conditions where here when you grow this particular yeast cell called Pichia pastoris in methanol, it makes these giant peroxisomes. It requires this peroxisome because it contains an enzyme that can degrade methanol, and so that provide nourishment to the cell. So cells can grow in low quantity of, it's almost like getting drunk in methanol, but at a very low concentration. Okay. And by this process, big peroxisomes are made. When you move these cells to glucose medium, when there is no methanol, then these peroxisomes are not needed. And therefore, autophagy, when they'll degrade these peroxisomes to get energy from all the proteins that it has made here, and it can be utilized here. And by moving them back and forth, one can study autophagy and also use them to identify drugs that are involved in either stimulating autophagy or inhibiting autophagy. Here is a list of compounds that we identified from almost more than 2 lakh compounds that we looked for. Among them, we have found a handful, and a few of them are represented here. These 4 compounds here actually promote autophagy. By adding these compounds onto the cells, we see that there is more and more autophagy. Okay, and if you add some of these 3 compounds here, you get to see that there is less and less autophagy. And so we have identified, therefore, like the genes that we identified in yeast, we can also identify molecules that can either induce autophagy or inhibit autophagy. Okay, and it's useful in various ways that you can use these small molecules as tools to understand how autophagy functions. Okay, so how do we? I'm not going to show you all the data here, but I'm going to summarize that we can now identify this molecule which actually begins. This is important for induction of autophagy. All the molecules you'll see are inducing autophagy. Whatever molecules appear here will be inhibiting autophagy. And the ones that induce autophagy up here are the ones that act at various stages. For example, this molecule azacitidine acts at a very, very early stage and induces signaling pathways that are important for autophagy induction. BAL11, for instance, prevents the closure of this autophagy. So it remains open, sort of. That's what we think. And as a result, it's incompletely formed autophagosomes. 6-bio, for example, we think it enhances fusion between autophagosome and lysosomes, therefore allowing more and more clearance of the cargo that it has captured in here. So EACC does exactly the opposite. It prevents this fusion, and by preventing this fusion, it is not letting autophagy complete. Autophagosomes are unable to degrade unless they get hydrolytic enzymes from the lysosomes, and therefore just having autophagosomes is not sufficient to clear the cargos. Okay, and so finally we have this molecule which acts at a very late stage, so that even if autolysosomes are formed, the hydrolytic enzymes are unable to degrade all this cargo because this compound can block that function, and that proteolytic cleavage function, because it's blocked, you don't see any degradation. So you can accumulation of autolysosomes happen in that particular case. In the first yeast picture that I was showing, there were accumulation of autolysosomes. Okay, so this is what we get to see. Sometimes people get very excited when we publish this paper where we show that this molecule is there. Companies started selling this, saying that, oh, here is an autophagy inhibitor identified in the lab. So you have EACC here, 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 here. All these companies now sell this, saying that, oh, this is a novel reversible inhibitor of autophagy flux, blah, blah, blah. And this is completely coming from our study. Okay, so there can be uses for studying this. So yeast gives us a lot of information about how autophagy can go forward, go slow, what genes can be involved, can we identify new molecules that are involved in this process, and so on. We will now look at part 2 of the talk, where we are looking at pathogens. And these are bacteria, viruses, and so on. And many of them will infect our cells. When they infect our cells, they start growing inside our cells. And there is sometimes not many ways of stopping them. And one of the ways of stopping them when they're growing inside our cells is to capture them and degrade them via lysosomes in a process known as autophagy. So that wing of autophagy that deals with pathogens, because pathogens come from outside, they're called as xenophagy. Xeno means foreign, and so any foreign particle coming from outside like bacteria and viruses can also be captured by autophagy and degraded. So I'm going to play you a small movie. This is a live cell microscopy, fluorescence microscopy of cells, where the green color is our autophagy. So this is one cell. When it plays, you can see this cell, and inside this there are 1, 2, 3, maybe 20, 30 bacteria, red colored bacteria. Okay, and green color is the autophagy machinery trying to capture each one of these bacteria and trying to stop it. Okay, let's see what happens when this movie is played. It's a movie over a period of 6 hours, but now we'll be playing them only in a few seconds. Okay, here it goes. You can now see that the bacteria start multiplying inside this HeLa cell. And when it is multiplying like this inside a HeLa cell and the whole cell seems to get filled with this bacteria, you can now start seeing autophagosomes going around these bacteria in some cases. Here, and these 2-3 places you get to see them. But the downside is the bacteria seem to overwhelm the autophagy machinery, and this doesn't seem to stop them really from multiplying. So what's happening here? Can we use our molecules to induce autophagy to see what happens? When my student Veena tried to address this problem, she found out a very interesting observation. Okay, she found out that these are bacteria here in our cells. Here some bacteria. Here is one cluster of bacteria, and here in some cluster of bacteria. Wherever there are these clusters of bacteria, these red colors are lysosomes. They were not close enough onto this bacteria. As a result, unless these lysosomes come close enough to this bacteria, they will not be killed. They were only plentiful present in those cells where there was not much bacteria. Okay, but when we identified a compound called azacitidine and treated these cells, you could now see that wherever there was bacteria, we could get more and more lysosomes coming there and trying to kill those bacteria. And I think that this compound works by elevating lysosomal levels and allowing more and more capture of these bacteria and fusing them with lysosomes so that they can be cleared and degraded. So this is another novel way one can think about killing bacteria inside our cells. So antibiotics directly work on bacteria and kill them. But in this case, we are looking at compounds that perhaps do not directly target the bacteria, but tell our cells to increase lysosomes or go and capture the bacteria better, and so that we become sort of immune, our immunity sort of increases so that we can directly address them. The advantage of doing some of this kind of work is that irrespective of the pathogen, sometimes we may be lucky enough to identify small molecules like this azacitidine that can be effective against different kinds of bacteria. Sometimes antibiotics only work against some class of bacteria, but if you have a good innate immunity built up inside a cell to look out for several such pathogens, then maybe it will be very effective. So this is the message that I wanted to give you guys today, that apart from using antibiotics, which can lead to drug resistance, you can have such innate immunity boosters that can then help clear the bacteria by inducing autophagy and by performing xenophagy. Okay, so here is how we think the compound works. The compound goes inside the nucleus, turns on a set of genes by using a transcription factor called TFEB, which is generally sitting on lysosomes. Okay, this is the mechanism, and by which all the autophagy and lysosomal genes are expressed in much abundance, leading to lots of autophagy and lots of lysosomes present inside a cell. And they can then capture this bacteria which enters, and then they can be cleared with lysosomes. Okay, that's how we think the mechanism is. Okay, how does this work? We did this in Petri dishes, but does it work in mice as well? So in fact, we took mice and infected them with salmonella, working in collaboration with a company called Vipragen. And we can see here that in the liver, which is zoomed in here, we can now see the following. When we had infection, we could, when there was no infection, normally these red dots indicate how much autophagy is happening inside a cell. But when we had infection, the infection is smart enough to suppress autophagy so that it can survive, and you don't see much of these dots. Okay, but when we give this compound along with the infection, we see many more dots that are lighting up like this, suggesting that autophagy is induced in this, and this also resulted in increase in autophagy markers inside in the mice, which also concomitantly resulted in decrease of infection in these cells. Okay, we could see some decrease. So suggesting that this can be also a very powerful way of controlling infection in organisms. Okay, so in the last part of my talk, I will tell you how autophagy not only is useful in gobbling whatever intracellular materials as well as pathogens, but in some cases it can also capture protein aggregates and then clear this protein aggregate. So why are protein aggregates so important? It turns out that our cells are like a constant factory of making proteins. They keep making proteins at an enormous speed every second, actually. And then when these proteins are being made, they are being made for carrying out various cellular functions. It's possible that there could be some bad proteins inside our cells that tend to become sticky, and they start forming a clump. And these clumps then can attract other proteins and form bigger clumps. And this kind of a growth of clumps is very, very bad for the cell, and the cells will eventually die. And this is the way, we may lose a lot of cells in our body as we age. Especially when we age, our quality control mechanisms, including autophagy, goes down. And as a result, these kind of aggregates build up inside a cell. Unlike, for example, our skin cells, our brain cells are not continuously made again and again. If you lose your brain cell, it's likely that it may not be completely replaced. Okay, so if protein aggregates from our neuronal cells or neurons and they die, then slowly you will have effects that you see because of loss of such neurons. If the neurons are playing a role in controlling your movements, then you will see that you will be restricted in their movements. And therefore, neurodegenerative diseases have a close association with autophagy. Okay, so the idea is, can autophagy come to the rescue of such diseases, neurodegenerative diseases, Alzheimer's, Parkinson's, Huntington's, amyotrophic lateral sclerosis, spinal spiral ataxia, and so on. Okay. So here is a cartoon showing what happens to people with Parkinson's. One of the things you can see that their posture is very stooped. Okay. And their facial expression is not so great. They cannot speak very well. They cannot swallow food. They drool. Most important common feature everybody notices is that they tremors. Their hands shake, their body shake. They're unable to, move properly and take care of themselves, and it can become worse and worse. And one of the reasons that happens is that this is because in the cells you have this protein aggregate clumping in a region known as substantia nigra. So what happens, and what is this protein buildup, and where is substantia nigra? So for that, if you take a cross section of the human brain, this is how it will look. These are your eyes, and here is a small region which is highlighted in here, which is called substantia nigra. Nigra means it's colored, and you can see this for this colored region here, dark colored. That's called substantia nigra. In this substantia nigra, there are neurons known as dopaminergic neurons. Dopaminergic neurons, of course, provide dopamine, and they are important for muscle control. So all your movements, in some sense, are reflected of the activity and healthiness and function of these neurons in substantia nigra. And they die during Parkinson's. And in Parkinson's disease, you can see it's very, very faintly colored, meaning by most of these cells that were there, they are not there anymore. And therefore, your muscle movement is severely affected. How can that study be done? We can't go and look at everybody's brain and study that. So what we do, it turns out that these brain cells die because of protein aggregates. And these protein aggregates can also be introduced in a humble yeast cell as well. And so what you see here is a single yeast cell carrying alpha-synuclein protein, which is fused to a fluorescent protein known as EGFP or GFP. And these cells now also form such massive protein clumps. Okay. And like the way these protein clumps kill neurons, alpha-synuclein, when expressed in yeast cells, will also kill the yeast cells. It is interesting to note that yeast cells do not have alpha-synuclein, but you can, using recombinant DNA technology, you can introduce alpha-synuclein in yeast cells and study these yeast cells, how they are dying, and can we save them? Because it is much easier to do that way than getting neurons from human brains and things like that. Although people keep trying doing that, but for doing a lot of experiment, this is an easier strategy. And while doing this, you see that cells die, but you can also ask, can I identify some molecule that can induce autophagy to capture these aggregates, bind to them, and clear them like this, so that in the lysosome, if it gets cleared, then the cells will be healthy and have a proper distribution of alpha-synuclein that we see. And why do we do this approach? The approach is very simple, that you can take these yeast cells and ask, are there any molecules that you can do through a big screen like this that can induce autophagy? And that part of autophagy which deals with clearing of these black aggregates are known as agriphagy. Like xenophagy was foreign, agriphagy is an autophagy wing that deals with clearance of aggregated proteins. Okay. And if you identify a very interesting molecule that can clear aggregates in yeast, and because autophagy is a very conserved process, you can ask whether autophagy now that happens in mammalian cells is also influenced by such drugs. So you can induce autophagy in mammalian cells as well. And the ones that work, when the molecules that show induction common for yeast as well as mammalian cells, and by clearing this aggregates, then you can ask, well, if it is so promising, can we try it in mouse model? So you can induce aggregates in brain of the mice and then see if the molecule is any beneficial. Okay, so I'm going to cut out all this data and I'm going to quickly show you how we did the screen. So normally, if you do a growth curve, we can write down this for some people who don't know. I'm showing very little data here, but if you do a growth curve, the yeast cells will grow like this, like sigmoidal curve, right? But if they have the aggregates and they're dying, they become like this. They don't sort of grow very well. But then you can have molecules like this X molecule, does nothing to the cell. There are still aggregates and they just sort of die away. But once in a while, you may get some molecule like Y that has cleared all these aggregates in the lysosomes and then has proper distribution of alpha-synuclein on the plasma membrane as it is seen for yeast. Okay. And so you can see, you can do this kind of screening in small plates, and you can screen hundreds and thousands of compounds and ask if you find any molecule like Y that does this. Okay. And then you do a lot of testing. And finally, if you're very, very convinced, you can then take it to the mouse model. And when you go to the mouse model, you can do, you can give them a toxin called as MPTP. So what you're seeing here is the brain sections of the mice, like 4 sections are here. And you can see for the normal mice, 5 sections here, one of them is zoomed in here. If you go to this area called substantia nigra pars compacta, you can see some stained region here. And this stained region indicates the dopaminergic neurons there. But in the drug treated, which is the MPTP treated mice, you can see the substantia nigra pars compacta is cleared up. Okay, and suggesting that these neurons are most likely killed when you give this toxin to the cells. But if you give any of the molecules that we have identified along with this toxin, you can see that there is not much of cell death, and suggesting that maybe the neurons did not die. And that's what we can count them. Generally, there are around 10,000 of such neurons, and they can die when you do it. 1/4 of them, more than 1/4 of them are gone. And when you give the compound, you can see some effect of this compound. You can zoom in this area and look at the cells, how they're looking, are they looking healthy or not? And they seem to have good amount of this stain, indicating that the cells are there. Whereas in MPTP, you get a very poor stain. Okay, the compound seems to save the cells, but how is it doing? When we look closer under the microscope, you can see that these cells, which are the dopaminergic neurons of substantia nigra, have lots of this green dots, suggesting that they are doing a lot of autophagy. Okay, you can count these dots in number of cells and count them versus the untreated ones. You will know that the compound treated ones seems to do a lot of autophagy. And these neurons, when they are doing autophagy, you can also count the number of protein aggregates that are there. So these white dots here are the protein aggregates. So the ones that did not get the compound will show a lot of such protein aggregates. And the ones that got the compound did not so much show that much protein aggregates, and they also showed increased autophagy. Okay, you can quantitate that. The data I'm not showing here. I just wanted to show you how the data looks when we do these experiments. And finally, the biggest question is, Are their muscle movement improved, right? All that is fine, but how are these mice doing? So this is Shashank. He's now a postdoc in UK, but he did this experiment several years back. And what you'll see here are these mice. So these mice, normally the mice are extremely well trained in rotating, balancing themselves and running around very small crevices. Okay. So normally the mice, if you put a slow speed and let this rotate, they will nicely balance on it. And below this, we have not shown here, but we keep a nice sponge cushion on them so that if they fall, nothing happens to them. So all the mice generally will not have any problem. But when you see a Parkinson mice, see, when we start this, you see a Parkinson mice is unable to grip or even walk on the rotating rod. This is called a rotating rod behavior test, okay, mice behavior test. And you can see that we are giving them multiple opportunities to balance on it, but either they get tired or they're unable to keep up with the slow speed of this rotating rod, and they generally just fall down. Okay. And here is the same guy who got the compound. Okay. He is almost like wild type mice. He doesn't seem to really have any problems moving, and he was given the same toxin as others. Okay. And so he tends to survive better. So what it tells you that you can quantitate this. You can say latency to fall in seconds. So in the control, you can see most of them are nicely balancing. But when you give them this toxin to knock out the substantia nigra pars compacta dopaminergic neurons, you can see most of them are unable to balance. But if you give them the compound, autophagy inducing compound along with this, they are able to nicely balance, suggesting that based on all the data, that they are able to balance and they don't have motor coordination very good, comparable with that of the control. Okay. And I have not shown you another test. It's called open field exploration test. You put mice in a box. It's generally very curious. So it starts running around like crazy. You can put a camera and you can see in 5 minutes how much it ran, the distance it covered in, let's say, 5 minutes. That also, if you plot MPTP people, mice, because they have problems in motor coordination, they're unable to run as much. And as a result, they will be slow in their movement and will not cover the distance that the control mice covers, right? And that's what is reflecting here, that the mice that do not have a good dopaminergic neurons in their substantia nigra have unable to move as much. But if you, along with this toxin, if you give the compound, they seem to be doing fairly well. Okay, similar to that of the control mice, suggesting that their neurons are there, they are functional, and the mice have no, at least using these two tests, do not seem to have any problems moving around. All this is very good, but you should also know that when this compound was given, the neurons died, right? So if you wait long enough for the neurons to die and then give our compound, because it's possible that in real patients, many of them may have lost a lot of neurons and already have tremors. So in such cases, what do we do, right? So we tried seeing if that works, and unfortunately, the compound doesn't work that way. If you wait long enough for the neurons in the brain to die and then give your compound, of course, because there are no neurons now, those neurons doing autophagy is out of the question, right? And therefore, these mice do not, in spite of getting the compound, when they're co-injected, they show effect. But post MPTP injection, if you wait long enough, 48 hours, and then give, they do not. They fail. The compound fails, suggesting that an early intervention is required for autophagy. Of course, if a lot of neurons are dead and are non-functional, then it may not be possible to rescue using this approach. Okay, so this is the lesson that we learned, some successes and some failures to understand how autophagy happens. So in the next slide, I'm going to summarize what we understand and how autophagy helps in clearing these aggregates. Okay, so this is going to be next 2 minutes. Just try and focus and see if you can understand this concept. So normally cells have aggregates. All our cells produce some aggregates. If they're not taken care of, they form bigger aggregates and they're very dangerous. Okay, but normally aggregates do form the cells. When the aggregates form in the cells, they are generally coated with another protein called ubiquitin, and then which subsequently with P62, this act like an eat me signal for autophagy to come and capture them. So this autophagy will come and capture them like this, and this autophagosome can now sequester this entire aggregate away from the cytoplasm, so it cannot cause more damage. However, it cannot degrade these aggregates because it doesn't have hydrolytic enzymes, which it needs from lysosomes. Okay, so it can capture everything like this, but unless there are lysosomes, it cannot go into it. But when there are lysosomes available and autophagosomes have captured it, what autophagosomes do is to go and fuse with lysosomes so that it clears the aggregates. And this is what happens. So you can have many of these autolysosomes formed and they'll come and degrade. This is what is happening normally. But in neurodegenerative conditions, there are more and more aggregates, much more than what the autophagosomes and lysosome system can handle. As a result, the aggregates seem to be winning the battle, and they will be always, more aggregates inside a cell. What we think happens with one of the molecules that we have is that this molecule makes, in presence of this autophagy inducer, it makes these autophagosomes capable of fusing more and more with lysosomes like that. It encourages fusion of these autophagosomes with lysosomes. And as a result, there is enhanced autophagosome-lysosome fusion, resulting in much more clearance of the aggregates than what is normally possible. And this leads to something called as the protective function. Cytoprotection means protection of the cells, the cytoplasm, as well as neuroprotection, suggesting the neurons do not die. Okay, so I'm going to conclude here. It was a lot of work, and I summarized it here. And sometimes when you are lucky, you in a good journal which is published by the Lancet, they can also put our work in the cover page, saying that we have identified a molecule that can, like a vacuum cleaner, suck up all the aggregates and clear the brain in cases of Parkinson's disease. Okay, so in the lab we have many groups. We have an yeast group, we have the xenophagy group, and some agriphagy group, and there are other stories which I did not tell you. And without great collaborators, it's not possible, I think, to do great research sometimes. And so I'm lucky to have really good people with me. Professor James Chalaya, who helped me with all the mice work. He's at the same institute where I am. And actually, he comes to Trichy often. And he's from Madurai, an excellent scientist, neuroscientist. Okay, I am also benefited by collaborating with Professor Falguni Aladi. She's in NIMHANS, Bangalore, and she's also a Parkinson and amyotrophic lateral sclerosis expert with whom we deal with. Other work I was not able to talk to you today, but these are some of the other people with whom we are working to do it. All this work also requires a bit of money, and so it came from Wellcome Trust and my parent organization CSIR and other funding agencies like DBT, DST, and DRDO. And I have 6 PhD students who have graduated so far, and 7 of them are there. And like each one of them, when they graduate, we call them ATG. So Piyush is ATG 1, Suresh is ATG 2, Sunena is ATG 3, Gaurav ATG 4, Soumya ATG 5, and Veena ATG 6. And we have few of these guys going to graduate soon. So we'll wait and see. So I hope I have not put you to sleep. At this point of time, I'd like to thank you for your attention, and I'll be happy to take any questions if you have. Thank you.
Thank you for your informative session, sir. It's time for queries. Students, if you have any queries, you can ask for it. I repeat, it's time for queries. If students have any queries, you can ask for it.
Sir, am I audible?
Yes, please go ahead.
Yes, sir. Sir, you have shown in HeLa cell lines, you have infected with Salmonella. So in that, with the autophagy enhancer, it can kill the bacteria, right?
That's right.
My question is, if nowadays all the antibiotics which is available are resistant to almost all the, against all the microorganisms. So is this idea is useful one for that cases, for any multi-drug resistance organisms? Can we suggest these ideas? That means you have shown 4 autophagy enhancers. So can we think in that way? Is it ideal one or?
Yeah, there is no easy answer. Very good question. This I get asked often. See, antibiotics have an advantage that they can be directly acted upon on the bacteria and kill it. Okay. Whereas here you have to capture them, take them to lysosomes. It's a longish process. And we right now don't know how much infection, how much autophagy, how they can be controlled. Okay. while antibiotics has almost now, I don't know, since Alexander Fleming's discovery, since the World War time, we had the benefit of more than 60, 70 years worth of research to really understand how antibiotics work and what is their potential. And whereas this field is very, very, very new. Okay. And so I think we'll have to give it a little bit more time to see there is real potential that we can have so much autophagy that every bacteria that enters or bad bacteria that enters our body can be cleared. But I don't see, in fact, what I would see is that in the future, maybe there will be a combination of antibiotics along with this kind of autophagy induction. Maybe that may be beneficial, where your innate immunity is boosted enough to really help clear the bacteria along with the antibiotic effectiveness. But as you say, if there are totally antibiotic resistant bacteria and there is no other way of stopping it, I think those will be the real test for autophagy inducers. And whether the molecules that we have are good or not, or should we look for more, only time will tell.
Okay, sir. Thank you, sir.
Thank you.
For it. I repeat, participants, if you have any queries, you can ask for it. Thank you so much, sir. The session was very informative.
Thank you. Thank you very much for having me. Bye.
Dear participants, I kindly request you all to fill the feedback form for your valuable feedback, which is being posted in the chat box. I repeat, dear participants, kindly fill the feedback form for your valuable feedback, which is being posted in the chat box.
We welcome the dignitaries to BICPAC' 2022. Welcome you all. Kindly be seated. To improve the golden moment of opportunity and catch the good that is in our reach is the great art of life. A scintillating good evening to one and all present here. We graciously welcome you all to the valedictory function of the International Virtual Conference on Biological Innovations and Computational Exploration for Pandemic Challenges, BICPAC' 2022.
The function of prayer is not to influence God, but rather to change the nature of the one who prays. Before we advance into this momentous instant, let's instigate the holy presence of God and march towards an extra mile with a smile in the task of molding young minds. I request everyone to stand up to extend our prayers to the Almighty. I invite Dr. M.G. Daniel Andrew, Assistant Professor, Department of Biotechnology and Bioinformatics, to lead us in prayer. Let us all rise to seek the shouts of heavenly joy.
Every eye closed and every head bowed. Our dear and gracious, loving Heavenly Father, we thank you for all of your support, your direction, your guidance in the successful conduct of this two-day international conference, BICPAC' 2022. Lord, we thank you for all of your mercies toward us, toward the Department of Biotechnology and Bioinformatics, Bishop Heber College, for the ability to organize such a marvelous conference. We thank you that our hard work and our efforts have paid off, and we are sure that we will reap rich dividends in the days to come. We thank you for all the speakers who gave their expert talks. We thank you for all the delegates, the participants from across the country. We thank you for the speakers, both national and international. We thank you for the hard work of the organizing committee, the organizing secretaries, the convener, the co-convener, and for all the dignitaries who are present on the dais right now. Father, we thank you for your benevolence, your goodness, your mercy, which is matchless, and we will sing of your mercies and your love. We give you all the praise and the glory in the precious name of our Lord Jesus Christ, we pray. Amen.
Thank you, sir. Let's all remain standing. The departmental choir will lead us in the prayer song further. I request everyone to please be seated. I thank the choir for the pleasing song. If the sign on your heart says welcome, the love will come pouring it from everywhere. Moving forward, I would like to invite Dr. Bliss Ramya June, Assistant Professor, Department of Biotechnology and Bioinformatics, to welcome the gathering with great zest and enthusiasm. Please, ma'am.
Good evening to everybody. The Bible says, "For the Lord giveth wisdom, out of his mouth cometh knowledge and understanding." Ladies and gentlemen, on behalf of Department of Biotechnology and Bioinformatics, organizing committee members of this international virtual conference, BICPAC 2022, I consider it a privilege to welcome such successful, renowned, humble person. I extend a warm welcome to our respected chief guest of today's valedictory function, Dr. C. Ananda Ramakrishnan, Director, National Institute of Food Technology, Entrepreneurship and Management, Ministry of Food Processing Industries, Government of India, Tanjore. We are pleased that you accepted our invitation and has come here to take part in this function amidst your busy schedule. Welcome you, sir. Man with energy, enthusiasm, focus with immense vision, leading our institution to greater heights. I am very much privileged to welcome our dynamic principal, Dr. D. Pal Dayabaran, to grace this occasion, which is going to be the grand one. We welcome you, sir. It's my immense pleasure to welcome our Dean of Extension Activities, Professor Dr. V. Anand Gideon, Associate Professor and Head, Department of Botany. We welcome you, sir. I welcome our Dean of International Relations, Professor Dr. R. Rajkumar, Associate Professor, Computer Science and Head of Data Sciences. We welcome you to this valedictory function, sir. I wholeheartedly welcome our Head of the Department and convener of this international conference, Professor Dr. Violet Dayabaran, joining with us in virtual. She's a lovable person. She always cares, guides, and prays for the well-being of the Department of Biotechnology and Bioinformatics. We will welcome you, ma'am. I am very much privileged to welcome our UG head and co-convener of this international virtual conference, Professor Dr. Jasmine. She is a dedicated, loyal, humble person in our Department of Biotechnology and Bioinformatics, leading us in an effective way. I am very much happy to welcome our young and energetic organizing secretaries of this International Virtual Conference, Professor Mr. J. Dinesh Raja and Professor Dr. M. Vinith. Their contributions towards this International Virtual Conference is tremendous, which carries out a milestone to our Department of Biotechnology and Bioinformatics. We welcome you, sir. I extend my warm welcome to all the eminent scientists, scholars, student communities joining in this virtual for this valedictory function. I am happy to welcome my dear colleagues and non-teaching staffs to grace this occasion. Once again, I welcome all to this grand valedictory function to this International Virtual Conference, BICPAC 2022. Thank you, one and all.
Thank you, ma'am, for the warm welcome. No person was ever honored for what he received. Honor has been the reward for what he gave. We are profusely elated to take an opportunity to honor our guest of the day with great joy and immense exultation. I request our beloved principal, Dr. D. Paul Dayabaran, to honor our chief guest, Dr. C. Anantaramakrishnan, Director at National Institute of Food and Technology.
third years, you were here only in the first 8 months of the first year. Again, 4 summers, you were not here. If you take second PG, you were not here last year. First PG, you are new. Is it okay? So all 5 years, it never happened in the past. Probably will not happen again till the world exists. Anyway, but we had to come. I told the yesterday in presidential address, we should not succumb to any kind of pressure. Is it okay? It's a kind of pressure. Problems are always giving you pressure. Normally, many of us succumb to pressures and problems. It should not happen. Is it okay? As students of Bishop Weber College, as students of higher education institution, whoever enters Bishop Weber or any other higher education institutes should come out with flying colors. Is it okay? You must take, of course, you rightly named the title of this virtual conference as Biological Innovations and Computational Exploration for Pandemic Challenges. Okay, a lot of innovation would have come. You explode and you've got innovative ideas. Thereby, many good things will happen. Is it okay? Even yesterday, we were talking about AIDS disease has been prevailing for many decades. Is it okay? But still, vaccines, suitable, appropriate vaccines, is not found. But whereas this pandemic affected the entire globe, from east to west, from south to north, all worked hard, and we have got a variety of vaccines. Thereby, we've been saved now. Is it okay? Maybe, of course, we suffered for the past 2 years, 2.5 years, but we are saved now. So, the more the problem, the more we work. Thereby, we succeed. Is it okay? So, dear friends, always as students, we have to work very hard. Just now, we're talking with the chief guest in my room. He's coming from a reputed institute of national importance. Is it okay? So, and they provide internship for students of, let us say, BTech food processing, MTech, like that. You know, you're doing MSc Biotechnology, you're doing MSc 5-year Bioinformatics. Is it okay? For internship, they normally, no, we have to invest money for better things. Is it okay? You know how much school fees? If you join a G school here, you have to spend a lakh for 1 year. Is it okay? If you spend 80,000 rupees in Bishop Weber, you can get master's degree in data science, master's in computer science, master's in MSc Biotechnology. Is it okay? Just 80,000 rupees. Is it okay? Whereas for school, you know, I know some of the student friends now, first years, when I asked, did you submit the TC or mark sheet? They said, no. So give me some time, sir. We did not clear the dues in the matriculation school. They said, they know your parents may be ordinary financially, hoping, they said, okay, somehow I managed to pay my students, my sons, my wards, my daughters to study in a good school. So they admit, but they are unable to pay the school fee. Is it okay? So therefore, they get into trouble. But here, it's only we get quality education for a nominal rate, isn't it? So therefore, here that said, they charge 25,000 rupees for useful internship for 3 months. Is it okay? So you should not think to spend money for your betterment. It's kind of an offertory. When you go to a temple, when you go to church, we put offertory. You should consider payment of fees for education as an offertory. This offertory is used for paying salary to your gurus, your faculty members. We get fees from 12,500 students. We give scholarship for 1,500 students. Your money is given to poor student deserving a scholarship. So you must have that kind of mind. We can come. Is it okay? In those days, playing county cricket in England was a great achievement. Couple played for Nottinghamshire. Is it okay like that? You know, Sachin played for one county. Now, at the time, Indian cricketers were considered as ordinary players. Is it okay? They work very, very hard. The past 3 decades, when I was doing my +1/+2 in late 80s, late 70s, 78, 80, at the time, you know, in England, India used to struggle when they go to Pakistan, when they go to England. Now, all the players are coming down to India to play for IPL. Is it like IPL? How it happened? They work very, very hard, lot of training sessions. Is it okay? The club is inviting a foreign coach. They come and coach our players. So when you work very hard, you can transform into a valuable product. Is it okay? For valuable product, you have to become a valuable product, you have to spend some resources. Resources, sometimes it may be money. I mean, for a, let us say, if an academic topper, they said, okay, college may can give 15,000 rupees as a scholarship to attend internship, where thereby you can spend only 10,000. That is also possible. I said, okay, so dear friends, you may be an introvert, quiet boy. You may be a talkative fellow, but you must have a passion in your heart. You should come up in life. Spending 3 years here, spending 5 years here, when you go out, we should be a transformed person. This is okay. Obviously, after 2 days, attending to all the sessions, you must be charged. You must be a transformed man. Other day, we had the chief guest, as the Vice Chancellor of Central University of Tiruvallur. He was formerly Vice Chancellor of MK University, formerly head of the Department of Biotechnology of our university. You're meeting him. We spoke almost for an hour. Now, sorry, is here, he will speak. So all this will charge you. We have always some mental blocks. Can I do? Can I do? If you can't do, then who can do? Is it okay? You must have that kind of attitude. So I don't want to take much time because we have our professors here to felicitate you and we have chief guests to address. So dear friends, once again, I appreciate and congratulate the entire team for their meticulous efforts for organizing this useful conference. I'm sure all the student friends are going to have a great time in the campus. Enjoy the campus. Enjoy your teachers, lovely teachers. Enjoy your friends, your wonderful friends among yourselves. Enjoy your friends.
Thank you, sir, for the pious chink of positivity. The fragrance of flowers spread only in the direction of the wind, but the goodness of a person spreads in all directions. We are extremely delighted to have with us Dr. V. Anand Gideon, Associate Professor and Head of Department of Botany and Dean of Extension Activities, to felicitate the gathering. Please, sir.
Good evening. Respected Principal, Honorable Chief Guest, Dr. C. Ananda Ramakrishnan, the Director of NIFTEM, Dr. Raj Kumar, the organizing secretaries, the co-convener, the faculty members of Department of Biotechnology and Bioinformatics, and my dear students. First of all, I would like to congratulate the Department of Biotechnology and Bioinformatics for choosing an apt topic, especially during this time. The challenges, the pandemic challenges, and where you have the biological innovations and computer exploration playing a major role in facing the challenges. For the past 3 years, to live itself became a challenge. உயிர் வாழ்றதே ஒரு சேலஞ்சா இருந்துச்சு ஆக்சுவலா இந்த 3 வருஷமா. We have all come across and come out with that challenge. Thanks to the technology, thanks to the sacrificial efforts taken by the scientific community, frontline workers, and all the scientific personnel behind making us live successfully. Challenges and struggles brings victory. Take up this as a challenge. For the past 3 years, the challenge that we faced, the challenge of surviving. Many who were with us earlier are not with us right now. But we are able to face the challenge, and then we are the survivors of the challenge. So this should teach us a lesson as to how to overcome all these challenges and struggles. Struggles brings victory, and challenges bring forth innovations. Innovations that should help human and other organisms to survive in a better way in this world. So take this as a challenge at this moment. And I once again thank the management of Bishop College, the principal, and the organizing team of Biotechnology for providing this opportunity. And my congratulations to the entire team. Thank you.
Thank you, sir, for sharing heaps of information. We are sure that your gripping message will definitely pitch a positive impact in the minds of young science enthusiasts. Science is organized knowledge. Wisdom is organized life. To keep up the momentum, I would like to humbly invite Dr. K. Rajkumar, Associate Professor, Coordinator of Data Science and Dean of International Relations, to felicitate the gathering with illuminating knowledge and experience. Please, sir.
Respected Chief Guest of this evening, beloved Principal, beloved Head of the Department of Biotech and Bioinformatics, beloved Dr. Anand Gideon, Head Department of Botany, dear Organizing Secretaries, Dr. Jasmine Ma'am, Mr. Dinesh and Mr. Vineet, and all the faculty members of Department of Biotechnology as well as Bioinformatics, and also faculty members who are connected online. Just now, Dinesh told me there are over 360 students connected throughout India across states, and some faculty members also are connected over Zoom right now, and many other students who are authors of the papers who submitted abstracts for the conference. Beloved students, bachelor's, master's students, and research scholars of Department of Biotech and Bioinformatics, very good evening, all of you. So I am immensely pleased and happy, in fact, to be here this evening and stand before you and to say few words about various aspects of the conference that you already completed for over 2 days. I was really thrilled when I looked at the title of the conference. It's really fascinating. So it speaks about the biological innovations as well as computational aspects of biology, biotechnology, and various kinds of biotechnology. And here we have our chief guest, Dr. Ananda Ramakrishnan sir, who is the director of NIFT EM, who is an eminent scientist and notable biotechnologist, I would say, especially food biotechnologist, well known across India. When we had a chat with him, he used to explain how students will get into NIFT AM through JEE as well as goes to do BTech as well as Master's program in food biotechnology. And now it has gone into various revolutions. Now it's going to be one of the top institutes of food biotechnology in India. So biological innovations plays very top role, I would say, just clubbing with the computational aspects. I had a chance to look at the various speakers who delivered talks over these 2 days. I was really amazed to see speakers across India. I could see one speaker from abroad, from Italy, I think Dr. Christina, and various well-known faculty members, scientists, professors from various institutes, including ISC, CSIR labs, University of Mumbai, and many well-known universities across India. So really so happy to see the Department of Biotechnology is doing a great service to the society, wherein bringing people from various states, connecting people. So, you know, this is the era of social media, so where everybody is connected everywhere. You have a lot of devices, Internet of things, Internet of devices, Internet of physical devices. So everywhere, everybody is connected, whether we know or not, and somebody is connected everywhere. So which helps to bring people into various, say, dimensions or various disciplines of related aspects of biotechnology. So I hope you had a great time over these 2 days, listening to the great lineup of speakers that you had. I have seen like there are 10 plenary speakers when I looked at the list. And also most of you would have a chance to present your papers orally. There were oral presentation sessions as well as poster presentation sessions as well. A very great lineup of thoughts. When I looked at the combination of biological innovations as well as computational explorations of different aspects, including the aspects of pandemic, and definitely you can think of education not just within the campus, but also across the campus. So this is something that's happening in the area of academics, education, as well as research. Those days have already gone. When I say somebody is a multidisciplinary department, you could have already seen that. Some colleges some students saying like is a multidisciplinary department or multidisciplinary program. So those days have already gone. It's not just a multidisciplinary program. We used to say those days like it's a multidisciplinary, right? So courses that we learn when we do bachelor's and master's studies. Then slowly you might have come across some more terms like interdisciplinary. So interdisciplinary education and interdisciplinary programs. So that could be considered as the next step to multidisciplinary education and multidisciplinary research. Now these terms are also obsolete now. Now nobody nowadays saying that. Nowadays is saying is a multidisciplinary educational system or interdisciplinary educational system. It's something more. Can you guess what is the current norm? What is the current terminology that is used in connection with multidisciplinary department or education or research or interdisciplinary department education or research? Can you guess? Yeah, now the norm in this, I would say, not this decade, even less than the decade, is transdisciplinary. So the transdisciplinary education is the norm of the day. It's the transdisciplinary research is a name or norm of the day. So when you look at, there are three different terminologies, okay, that are connected. You just Google and find out what could be the difference between interdisciplinary, multidisciplinary, and transdisciplinary. So as a department of biotechnology, as a college, and now we are looking forward to envision something that is transdisciplinary, departments, transdisciplinary education system, as well as transdisciplinary research. So without grabbing much of the time of our chief guest, so I congratulate, I wish, I thank everyone who are seated here, who are connected online, faculty members of biotechnology, faculty members of bioinformatics, distinguished professors who are connected online, and loving students who are all online, and all the people who are connected directly or indirectly to the department, like people from industry and everybody, stakeholders. Definitely, I wish this time could be a memorable time that could be for all of you. All the best. Thank you.
Thank you, sir, for your words of appreciation and encouragement. Words embody power to inspire or motivate us, but it is only we who have power to open up to see it and feel it. It's an optimistic sensation to hear the chronicles of achievements which promptly moves others. Let's set up ourselves to be driven towards progress by hearing the glimpses of achievements stacked by our eminent chief guest. I invite Dr. A. Sherlyn Rosita, Assistant Professor, Department of Biotechnology and Bioinformatics, to introduce our chief guest of the day. Please, ma'am.
Respected principal, honorable chief guest, dignitaries on the dais and off the dais, the scientific communities who have joined online, faculty members, research scholars, and my dear student friends, a warm greetings to all gathered for the valedictory ceremony. Live as if you were to die tomorrow. Learn as if you were to live forever. With the thoughtful quotes of Mahatma Gandhiji, it gives me immense pleasure and honor to introduce our respected chief guest, Dr. Ananda Ramakrishnan, Director, NIFTM Tanjavur. Dr. Ananda Ramakrishnan is presently the Director of the National Institute of Food Technology, Entrepreneurship and Management, Tanjavur. Sir was Principal Scientist and Coordinator for the Academy of Science and Innovative Research, ACCSIR, and CSIR CFTRI He is an active researcher with 2 decades of experience in research and administration. He obtained BTech in chemical engineering from AC Tech Anna University, Chennai, and completed MTech at Anna University, Chennai. He has done his doctoral research in chemical engineering with a specialization in food engineering at United Kingdom. His research endeavors are well documented in the form of 160 plus impact factor publications with an average impact factor of 5.17, 2 international patents and 7 Indian patents. He is also the author of editor 10 books and 68 book chapters. He has supervised 13 PhD theses. Dr. Ananda Ramakrishnan is the elected fellow of National Academy of Sciences, National Academy of Agriculture Sciences, fellow of Royal Society of Chemistry, Royal Society of Biology, Associate of Food Scientist and Technology, and the Institute of Engineers. He is a member of several professional committees of autonomous government bodies all over India. He also served as editorial board member for reputed journals and member of board of studies in various academic institutions. Dr. Ananda Ramakrishnan was awarded the prestigious ICAR Rafi Agamat Kidboy Award for Outstanding Research in Agricultural Sciences. He was awarded Tata Innovation Fellowship for 2019-20 by DBT, Government of India. He was also awarded the National Design Award 2019 for outstanding contribution by the Institute of Engineers, National Design and Research Forum. He is also the recipient of the prestigious Nasi Reliance Industry Platinum Jubilee Award 2018 and the AIFPA Special Platinum Jubilee Award for the Development of Food Processing Technology and Innovation 2019. He was also announced for the Tamil Nadu State Scientist Award 2018 by the Tamil Nadu State Council for Science and Technology. We thank you, sir, for kindly being our chief guest, and we fully appreciate you finding the time to be with us despite your tight schedule. Sir, it's a great honor to have you as a chief guest speaker for the valedictory ceremony of BICPAC'22. It's my indeed privilege to welcome Dr. Ananda Ramakrishnan for the valedictory address. Thank you very much for the opportunity. Thank you.
Thank you, ma'am, for briefing us about the impressive credential of our chief guest. Without any delay, let's dive deep into the ocean of innovation and feed our brain with immense knowledge. We are pleased to invite our chief guest, Dr. C. Ananta Ramakrishnan, Director at National Institute of Food and Technology, Entrepreneurship and Management, Ministry of Food Processing Industries.
Thank you, ma'am. Thank you so much. And it is wonderful to meet everyone. And nation is celebrating a science week this week. And I came to know Bishop College, also one of the center to celebrate our National Science Day. And respected principal and all the dignitaries on the dais and organizers of this wonderful event. And after a long time attending for the one physical meeting, when your department colleague came to our institute, I was too mind to accept or not, because I know Friday Eve, normally we have a lot of meetings, but he told, sir, our students are waiting for you to meet you. Then I thought of after a long time I'm meeting some students physically. Normally I used to give every day one lecture. Last almost, I never stopped my giving the lecture last 200 days. So today I thought of to meet you everyone physically. Thank you so much for giving the opportunity. When we are celebrating the Science Day, we have to remember our wonderful scientists who built this wonderful nation. People have forgotten a lot of scientists who contributed this nation to build such a level. It starts with our Sir C.V. Raman. The person, he never asked for I need this much funding to do research. I want this much space, nothing. Just he is sitting in his lab, doing day and night research. And he wanted to make impression in the world. India has the good science than anyone. That's what he used to do in his research. You remember that when Government of India wanted to give a Bharat Ratna award, he was told, no, I have to spend time with my student. That is the way the contribution made to the Indian science. So many people after that, they built the scientific content of our country. if you remember 1960s, we used to import huge amount of even rice. That time, central minister is C. Subramaniam. He was the agriculture and food minister. He called our Professor M. S. Swaminathan, that time he was the IRI director, how to sort it out this problem. Because India is not that economically strong country on that time. So he decided Norman style of breeding different varieties of the wheat can yield per hectare more rice as well as wheat. That's what Green Revolution has started. Today, 2 weeks back, Government of India announced we produced record number of rice and wheat, all the grains. 380 million tons of the wheat and rice has been produced. Think about it. If it is not here, what we do? We have to import. Already petrol import. If one war has happened, immediately price will increase. Think about every day your rice and wheat price has been fixed. Very difficult to live in our country. The scientific contribution made by our agriculture scientists, all the food engineers, including our farmers, they put enormous effort to make totally independent, sustainable country. Not only that, this year we even exported rice to other countries. That is the strength of science. Maybe the people are not realize the science well when what the title says, when things like a pandemic, people will look at science, not a God. God is always there, but scientist is the more than that, what I rated. So you are the future scientist. Think like that. Mankind can save by the scientist. That day, even first pandemic started, we don't know what is that. This COVID is the very serious or how to handle it itself. You remember that everybody is talking about ventilators. But now no one is talking because the science is evolved itself. Even SARS-CoV-2 or whatever it is now, the different variant, it takes some time. The predictive modeling, everything has been put up. The scientists worked hard. SAR has told about vaccines. Whatever the vaccines has come for AstraZeneca from the UK and combination with the our Cambridge University and India, but indigenous vaccine also developed in the same time, Covaxin. The owner of Covaxin, Krishna Ella, he studied in Tamil Nadu Agriculture University. He's an agriculture scientist. He started a company for different purpose. Later, he realized we can make vaccines for this COVID. He has taken the government support, DBT, CSIR, all the government agency put together. They worked very hard to make such type of attempt co-vaccines. Maybe aware of that everywhere discrimination is there, even science nowhere we can't leave that out. Still we are not get that note from other countries, but still co-vaccine is the one of the best vaccines what we produce from our country and we exported most of the countries. That is the scientific element, it's there. So science is a wonderful thing to do in mankind. One more scientist, Dr. V. Subramaniam. He was the biological scientist in the IIC Bangalore. When Government of India wanted to make it a one food research center in our country, they asked him to go and start the research institution in the Mysore, that's called the CFTRI. They given only a palace to start the institute. He went there, he sat, worked very hard to make the wonderful institute of Central Food Technological Research Institute. Now 50 years over, the contribution of that institute is in everywhere. Late 69, we used to import all weaning foods or baby foods. We don't have even single food product in ourselves. Why? India, we have mostly on the buffalo than the cow. Now things are changed. So myth was you can't convert buffalo milk into the milk powder. So Dr. V. Subramaniam has taken a challenge. He asked the army to provide the lathe because no fund from Government of India. So old lathe, he moved into the CFTRI. He constructed that lathe, the drum dryer. He created the powder from the buffalo milk. The whole world was laughing at India. They are telling, you can't make baby foods, but we produced that's called Amul spray. Till it is there, and how many, even you and me, everybody in the small kids, we used to take no different form of that Amul spray has evolved itself. But the time and energy, totally 12 people day and night worked in that lab. Still that spray dryer and the drum dryer existed in CFTRA. So how many of us we remember that, who invented that, how much time? Nobody claimed, he didn't patent it. For our country, he made it and he proved, and we made it proudly to say, yes, India can do anything, what you do that. That's what Amul has come. Amul itself is a biggest story. Professor Kurian was the Amul MD. See, milk is a different candidate. After maximum 3 hours to 4 hours, you can keep it after your collection of the milk. So how to store? Instead of transporting into the another places, storing in the bulk is a very difficult. He realized, he put the collection center everywhere, each and every village. He made the cooperatives, they collected everything and do that. Today, Amul is one of the leading company in the world in the food processing, the milk sector. The scientist is the Korean. See, that's what our white revolution or green revolution. Today we are comfortably, we can sell it a rice 1 rupees in the PDS system. Everything it is there, but a lot of unanswered questions are there. Who is going to solve? Who are studying today in front of me, who is sitting? You people has to solve our nation's problem in the future. We could do whatever we able to do that. If I take the blood sample of everybody here, I know the 50% students are girl students. I am very much sure most of them are anemic. They may be knowing well. Why? Last 20, 30 years, we put a lot of program in our country. We still be unable to solve the anemic issues in our country. See, fortification is not only help to solve the problem. You add how much amount of the iron into your product, or you take the folic acid tablet, nothing will work because it absorbs only 10% in our body. Except selenium and vitamin C, most of the micronutrient absorbs less than 50%. How to enhance the absorption in our body? So that is the science, we need to move on to that. Our body conditions, absorption is very, very less. So how to do that? So that problem we have to solve. When I was in UK, was doing the PhD, then I wanted to settle there. Then I told, I think in January 1st week, same time, and then I called my father, I told I wanted to settle here. He said, you are not coming to Pongal, to hometown, you have to come for my death. That's what he said. That's what I returned back from UK, because he's such a patriotic person, and he told you have to serve for this nation. I came. The first problem I have taken as why I'm telling my story is, at least you can, it is easy to do the science and you can contribute whichever possible way to our nation. That's what I'm telling this. Came to CFTRA, again I joined back as a scientist. I took the first problem of this iron problem. Then I found he can make it a nanoparticle. We know that our hair diameter is 1 lakh nanometer. So what I'm talking about is the 100 to 200 nanometer size. Is it possible to create a such type of iron particles? Can you deliver from oral? Even you can't see from your eye how to create, how to deliver it. That is the huge challenge of the biological challenge from us. Being a chemical engineer, being a food engineer, how to do that? That's the one. And I made it the particle, but I can't able to test because human ethical clearance not allowed in our country for doing the nanoparticles. How to test in the human? Then I searched. That time, Netherlands, one TNO system was there, artificial human digestive system, oral, stomach, and intestine. I asked them, they told nanoparticle is difficult, and even though if you send, we will test. The charge is huge. I can't afford to do that because we don't have the funding for that. Then I started, why not? We can't create the similar type of system for our country. I started that journey around 2009, day and night, because understanding the digestive system itself, great challenge. So I could recreate again oral, stomach, and intestine. Stomach, we scan the MRI scan, convert a 2D image into 3D image, 3D printed stomach. I think first 3D printed stomach has been done by me, and it has been created. Right now, Asia's first artificial human digestive system is there in the Niftam Tanjavur. Even you can come and visit and see that. And we published more than 20 papers. Is far better than what is the existed in the Netherlands system. It not only gives the glycemic index, glycemic load, food disintegration kinetics. Just you add your food, either biscuit or anything, automatically it grinds, it goes to stomach, make the digestion, go to intestine, observe how it takes place. Everything you can see from your eye, you will get the clear results. You no need to touch any human being. So this helps not only for the science and also institute. Institute is making huge money using this system. But again, as my seniors, what they have done, I have not done the patent. Just I published each and every component of the system to the open source. Anybody can replicate the system. That's what one could think, what best we can contribute to our nation. And the institute, when I came in 2016, just from here, Perambalur district, a lot of farmers were struggling to sell the small onion. The price was very, very low. Normally, the small salad onion, you have to sell it at minimum above 10 to 15 rupees. Then only whatever money you put, you get the returns. That time people are like farmers are throwing the onion. Just I called all my students and my faculty. Today all are going to Perambalur. We need to sort it out our farmers issue. If this institute is existed here, if we are not able to solve our own farmers problem, what for we have to be here? Then I took the bus, we went to Setikulam village. I told them, talk to the farmers, talk to them, what the solution today evening we have to tell. 5 o'clock you have to come and tell me. If you are not giving the solution, I am not moving back bus to the Tanjavur. So students, faculty, everybody talking to the farmers and all, and they had a discussion. They came to me around 4 o'clock. Sir, we can make the peeling of the onion. That will give the value addition. We convert as the onion paste and powder and flaky. It's possible to do that. How many days it need? What is the budget? They told around 25 lakhs, sir. We can do it in the 100 days. Okay, come back. We'll return back to Tanjavur. I told every day morning 10 o'clock, all of us to assemble in one place. First, I will explain what is my contribution to that project. Everybody has to explain. Then they have to go to their work, no class, nothing. Within 20 days, we made the integrated onion processing unit in the institute itself. And we installed the whole unit in the Setikulam village. Still, it is running for the farmers. It is working very well. Now, students are using that unit for making the products. So what I wanted to say, the science can change the lives of the people. They can change, make people life better, make more people smile. That's what we have to think. What is our contribution to the science? So my dear students, please come to the scientific field. It is a wonderful field. No other place your name can withstand. If you go to the IT industry, any industry, name will be there as the manager or whatever it is. But you become a scientist, just you Google your name, Wikipedia put it your name page separate, Google Scholar give your full how much citations are there. What else you can do it in the earth? Life is one time, we don't know the second life is there or not, correct? So among that life, around 20 to 30 years, we will be served in the society. We can able to serve. Can we do better service to the society? Knowledge you are getting from Bishop College, wherever you're getting knowledge. What use of that knowledge? How to convert that knowledge for the betterment of the people? The Science Week, we'll take that pledge. We will make better scientists in the globe. We will solve not only India's problem, global problem also we can solve. That is the what. Now we have to take the challenge, and I hope more people will come forward to scientific world, do the research. Future is, I said, still we are unable to solve anemic problem. Tamil Nadu, almost 40% of the people are getting anemic. Recently released Niti Aayog data is alarming data. One of the richest state in the country, but still we are not good at our health index. We are lower than the Chhattisgarh and UP and Bihar. So we need to improve as quick as possible, because what is the food you are taking? You are not taking the healthier choice of the food. We need to change that one. So if you are taking, you wish to take, how to compact that one. That is the solution we need to bring. What is the solution for You go to your home, your father or mother, your grandpa, at least one tablet they're taking, is it not? How to solve this problem? That's what you have to think. And we need to put all the effort. We don't know, after 20 years or 10 years, one more pandemic may come. Are we prepared for that? So all this solution in your hand. Hope this type of conference and innovations help to move on that directions. I think the environment what provided by the Bishop College and what your parents are ask you to do science as your profession, please practice as a science as a profession, not for anything else. All the best to my dear student. Thank you so much for listening.
Thank you, sir. It was indeed a captivating speech. It's time to honor the editorial board members. I invite Dr. Shirley Elgal, Associate Professor, Department of Biotechnology and Bioinformatics, to take over the stage. Please, ma'am.
The roots of all goodness lies in the soil of appreciation for their hard work and goodness. I'm very grateful to express our sincere gratitude to the members of editorial committee who worked day and night behind the screen for the grand success of 2 days international conference, BICPAC' 2022. It's a time to honor them. Now I request our honorable chief guest, Dr. Anantharavana Krishnan, Director, National Institute of Food Technology, Entrepreneurship and Management, Tanjo, to give away the certificates as a token of appreciation. I request Dr. R. Jasmine, Editor-in-Chief, BICPAC' 2022, to receive the honor. Now I invite the members of Associate Editors of BICPAC' 2022, Dr. T. William, Assistant Professor, Department of Biotechnology and Bioinformatics, Biotechnology, Bishop Weber College. Dr. Bliss Join, Assistant Professor, Department of Biotechnology, Bishop Weber College. Ms. S. Leelavati, Assistant Professor, Department of Biotechnology, Bishop Weber College. Dr. M. Jnanaraj, Assistant Professor, Department of Biotechnology, Bishop Weber College. Thank you, sir. Once again, I thank all the organizing members for their meticulous effort. Thank you.
We request organizing committee members to receive their certification for their excellent involvement in the International Virtual Conference. Dr. M. Rajadurai, Assistant Professor, Department of Biotechnology and Bioinformatics. Dr. B. Rajasudagar, Assistant Professor, Department of Biotechnology and Bioinformatics. Dr. S. Suetenamudan, Assistant Professor, Department of Biotechnology and Bioinformatics.
Now it's time to honor the judges of oral and poster presentation in BICPAC' 22 for their enthusiastic involvement. Dr. C. Cynthia, Associate Professor, Department of Biotechnology and Bioinformatics. Dr. N. Shirley Elgal, Associate Professor, Department of Biotechnology, Bioinformatics. Dr. K. Akila, Assistant Professor, Department of Biotechnology and Bioinformatics. Dr. R. Sharmila, Associate Professor, Department of Biotechnology and Bioinformatics. Mrs. A. Sherlyn Rosetta, Assistant Professor, Department of Biotechnology and Bioinformatics. Dr. M.G. Daniel Andrew, Assistant Professor, Department of Biotechnology and Bioinformatics. Dr. T. Jabaston, Assistant Professor, Department of Biotechnology and Bioinformatics. Ms. S. Leelavati, Assistant Professor, Department of Biotechnology and Bioinformatics. Dr. A. Anita Margaret, Assistant Professor, Department of Biotechnology and Bioinformatics. Thank you, dignitary. Bliss, ma'am, Assistant Professor, Department of Biotechnology and Bioinformatics. Thank you, dignitaries. Talent and skills are the crops that are nurtured, groomed, and given all possible opportunities to grow and bloom. This is a much awaited event in every student's life as it gives the returns to the efforts put in by them throughout their academic endeavor. It is a moment full of joy, a moment about pride and accomplishment, a moment of celebration. It gives a sense of achievement and a sense of responsibility towards fulfilling further accomplishment. It fills the heart with mixed feelings, fulfillment, anxiety, and eagerness. I humbly request Dr. T. William Raja, Assistant Professor, Department of Biotechnology and Bioinformatics, to take over the session for announcing the names of the winner. Please, sir.
Good evening, ladies and gentlemen. It gives me immense pleasure to read out the results of the most awaited part of the valedictory function. We appreciate all the participants who took part in the competition because participation is sometimes more than winning. It makes you a better winner for the next time. Yes, I'm happy to read out the winners of the oral presentation and poster presentation. The prize winners of each group of the oral presentations are as follows. Mr. Kamalesh from Rajalakshmi Engineering College, Chennai. Please clap your hands and appreciate the participants and the winners. Ms. Divya Rajaselvi from College of Veterinary and Animal Sciences. Ms. Akshada from University of Mysore. Dr. Sinos Sakariyachan from St. Pius X College, Kerala. Mr. Chetan Kumar from Kurukshetra University, Kurukshetra. From Rakesh, Mr. Rakesh from Christ University, Bangalore. Mr. Prima Carol from Kuvempu University. Mr. Maheshwaran from Jamal Muhammad College, Trichy. Dr. Grish from Maharani Science College for Women, Mysore. Dr. Nimisha from Central University of Kerala. Dr. Surendra Paul, ICAR, National Bureau of Agriculturally Important Microorganism, Uttar Pradesh. He is a scientist from ICAR CSIR Institute. Dr. Lavanya from University of Mysore. The best poster winners are as follows.
The journey of life and reminders of power of knowledge. As we come to the close of our valedictory ceremony, I invite Dr. Dinesh Raja, Organizing Secretary, BICPAC'22, to express the words of gratitude. Please, sir.
Respected Director of NIFTM, Tanjavur, Ministry of Food Processing Industries, Government of India, Honorable Principal of Bishop College, Dr. D. Baldayavan, sir, the Associate Professor and Head of the Mathematics Department, Honorable Dr. P. Anand Gideon, sir, Associate Professor and Head, Department of Botany, Honorable Dr. K. Rajkumar, sir, Associate Professor of Computer Science and Coordinator of Data Science Department, Honorable Convener and also the Head in charge of the Biotechnology and Bioinformatics Department, Dr. Jasmine ma'am, my co-organizing secretary, Dr. Vinith, our beloved members of the faculty, dear students, delegates from all over the nation, a warm greetings to you. On behalf of the organizing committee, we would like to thank you very much for your presence over here. At this juncture, I would like to thank our honorable chief guest, Dr. Ananda Ramakrishnan, sir, being a National Institute of Importance, how the director busy must be. The director has to deal with not only research and education. The director also interact with the ministry secretaries and also interact with the PM office. He's busy with every day. He's busy with a lot of commitments for the upliftment of the institution. Recently, the NIFTM have been conferred as a National Institute of Importance by the Parliament of India. So on behalf of the organizing committee, the Department of Biotechnology and Bioinformatics, I would like to thank you very much, sir, for your presence over here and deliver the valedictory address for our students. At this juncture, I would like to thank our honorable principal, sir. You know that our institution is a very big institution. And Tiruchirappalli, running the institution with 12,500 students with 500 plus faculty members is too difficult to run the institution. So in this busy schedule, I think we are celebrating a National Science Day week by the funding of the Department of Biotechnology, Government of India. So sir has to function with a lot of engagements all over all the day. So on behalf of the organizing committee, we'd like to thank you very much, dear principal sir, for your presence and guidance to run this event in an efficient manner. At this juncture, I would like to thank Dr. V. Anand Gudian sir, Associate Professor and Head Botany Department, and also my teacher. So thank you very much, sir, for your presence, and thank you very much for your kind support and guidance. And I must thank the presence of Dr. K. Rajkumar sir, Associate Professor of Computer Science and Coordinator of Data Sciences, for his valuable presence. Thank you very much, sir. And it is a time to thank the editorial board members. I think 300 and plus oral abstracts we received all over the nation. It is too difficult to compile. It is too difficult to receive feedback from all over the India. And we have to create rooms and we have to make them to give a slot. So it is too difficult. It's a time I have to thank with all my heart and all my soul, Dr. William sir and Dr. Bliss Ramya sir, ma'am, Dr. Leela ma'am, and Dr. Nyanda sir for their tireless effort to bring out the event in an efficient manner. Thank you, editorial board members. And also I would like to thank the editor-in-chief, Dr. Jasmine ma'am. It's a time to thank my faculty members. So my faculty members are experts in every field. Our faculty members are experts in publishing the papers. Our faculty members are experts in retrieving funds from different institutions. Our faculty members are efficient teachers. So they are the backbone of the department. So without my faculty members, it is not possible to run this event in an efficient manner. So I dedicate this conference to all of my faculty members to the Department of Biotechnology and Bioinformatics. And my dear students, you are the souls and you are the hearts of our life and everything. So the institution, the quality of the institution, the quality of the growth of the department is based on the students' feedback. So dear students, thank you for choosing Bishop Weber College as your place. You can have your bright future. Thank you, dear students, for choosing Bishop Weber College. And it's a time to thank the delegates from all over the nation. Thank you very much for acknowledging us. Thank you very much for your active participation in this 2 days international event. I thank Almighty for giving me the greatest opportunity to take my college, Bishop Weber College, and to take the Department of Biotechnology and Bioinformatics throughout the India and the globe. Thank you, Lord. Thank you, Jesus. And thank you, everyone. May God bless.
Thank you, sir. Time is short. Science is infinite. It's time to pull down the curtains of the International Virtual Conference, BICPAC' 2022.
Excuse me. Sorry, I forgot to thank our coordinator, madam. She's watching in online. He is the mother of our department. So whatever the thing which we are doing, so with her support and with her prayer, the department is growing in a way. I would like to thank our department coordinator, madam, Dr. V. Violet Dayabaran, Associate Professor and Head Dean of Sciences, for her wonderful support towards the betterment of the department always. Thank you very much, Dr. Violet, ma'am. Thank you very much.
Thank you, sir. Time is short. Science is infinite. It's time to pull down the curtains of the International Virtual Conference, BICPAC' 2022. On this note, let's conclude a remarkable, memorable, and knowledgeable international conference. On behalf of the Department of Biotechnology and Bioinformatics, we sincerely thank the dignitaries for being with us and enlightening us with your enthusiastic remarks. I share my heartfelt gratitude to the professors, office bearers, and students for their wholehearted support rendered in the success of this conference. We, the Department of Biotechnology and Bioinformatics, signing off. Thank you. Have a wonderful and fun-filled evening.
National anthem. Mother and motherland are greater than heaven. So to show devotion towards motherland, all of you please be ready for national anthem. Please rise for our national anthem.
अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल गंगा विंध्य हिमाचल यमुना गंगा उज्जल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मागे गाहे तव जय गादा जनगण मंगल नायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
I request the entire faculty members to kindly share the stage for a group photograph.